Good morning and welcome back to the very third day of the 10th annual NIAA Antibiotic Symposium. I'm Dr. Leah Dorman with Phibro Animal Health, and I'll be your co-host today along with Dr. Eric Moore from Norbrook. I just want to give a special thank you to our sponsors. Without them, this would not be possible. And those sponsors are the Beef Checkoff, As Though Leadership Institute, United Soybean Board, Norbrook, Phibro Animal Health, and the KC Animal Health Corridor. I also want to thank our planning committee. We had a great team this year: Dr. Eric Moore, Dr. Andy King, Dr. Megan Nichols, Dr. Heather Fowler, Dr. Paul Plummer, Dr. Michael Costan. Thanks to all of you. I know you put in a lot of hard work, time and dedication to the planning of this. So thank you very much for all your expertise and also for helping moderate the program. So just a quick review on where we've been the last 3 days. On Monday, we received some tips on how to communicate more effectively about antibiotic use and resistance. We hope that you'll take these tips that were given and use the key learnings from the last 2 days, as well as today, to help tell the story of responsible use in a way that really resonates with the listener. Yesterday, we heard updates on human health, on research and animal health, and that included the implementation of antibiotic stewardship for each major livestock species. I thought there were some really interesting things shared yesterday. There was certainly an overarching theme of continuous improvement, especially in the areas of monitoring, prevention, and stewardship. Today, we'll have the opportunity to learn more from an environmental and international perspective. But first, I'd like to introduce my co-chair, co-moderator, and comic relief, Dr. Eric Moore. And Eric, it's not every day I get to be the straight man, so thanks.
Yeah, I know, that's true. Thanks, Leah. Welcome, everybody. I'd like to give everybody a warm welcome. I think you have seen a great program so far, and we're going to continue with that today. I have the pleasure of introducing Paul Plummer. Dr. Paul Plummer is the Executive Director of the National Institute of Antimicrobial Resistance Research and Education, commonly known as NIAMRI. So located at Iowa State. He's also a professor and holds the Anderson Chair in Veterinary Sciences at the Department of Veterinary Diagnostic and Production Animal Medicine and Veterinary Microbiology and Preventative Medicine at the Iowa State University College of Veterinary Medicine. That's a lot of words, Paul, on there to have. But he's also been involved. This is the kind of Paul Plummer show because he was also a panelist yesterday. He's on the planning committee, and he's going to take us through a great set of speakers this morning. So with that, Dr. Paul Plummer, I'll turn it over to you.
Appreciate Eric and Leah. I agree. We've had 2 great days here, and we're excited to move to our next topic. So this morning, we're going to first hear from Jay Garland. And so let me tell you a little bit about this update from the Environmental Protection Agency and Dr. Garland. Dr. Garland joined the EPA's Office of Research and Development in 2011. He received his PhD in environmental sciences from the University of Virginia and spent over 20 years working on NASA's efforts to develop closed bioregenerative life support systems for extended human spaceflight. NASA recognized him for innovative technical achievements multiple times. My son would be very impressed with that, Dr. Garland. So, but since being at the EPA, he works on a range of topics, including methods for microbial community analysis, factors affecting survival of human-associated pathogens, and various biological approaches to recycling waste, hence that connection to NASA and the closed bioregenerative life support type of idea. His current efforts at EPA focus on advancing innovative approaches to water infrastructure, including decentralized water reuse and mitigating risk associated with antimicrobial resistance and water cycles. So without further ado, Dr. Garland.
Thank you, Paul. Hopefully you can hear me and
Yes, we can.
see me soon too, I guess. Hopefully I opened my camera up. Thank you for the introduction, Paul, and yeah, tell your son to call me anytime. My experience with NASA, I was very fortunate to have that in my career, but I'm also very glad to have returned to Earth in these last 10 years to work on some more terrestrial-based issues. And today I want to talk to you a little bit about kind of an early stages of planning work related to a NARMS effort to look at a surface water pilot study as part of NARMS. So, you know, a lot of times you really want to be able to present results, but you're not going to get results today. We're really talking about, we're early in the planning process and going to lay out kind of the issues for designing this pilot and the plans. And I think that's also while it's nice to present data, I think it's also good to engage stakeholders like yourselves early in the process. And hopefully this will start that conversation. Let's see. The challenge here is the complexity of dealing with antimicrobial resistance in the environment. I mean, I think this is obviously something that's very all of you are aware of. You know, I think from the perspective of an environmental contaminant, I think the unique and challenging aspects of AMR is that it's not just the attenuation of a given contaminant in the environment, but it's not only that, the attenuation of both the genes and the resistant pathogens themselves, but also potential transfer and amplification of those genes into non-target organisms in the environment. So I think that's definitely a challenge. The other unique aspect about AMR in the environment is that it is a naturally occurring contaminant, right? In fact, antibiotics were originally derived from soil microorganisms. So it's important to understand what the influence of the human environment is, the built infrastructure. And from this diagram, you can see there's a complexity of interactions there, both from agriculture and the built and directly from humans as well. And the built infrastructure in this case, especially with us in EPA, we do a lot of work in the wastewater systems of our nation. And in that case, that is an environment. As soon as it leaves the body, it's a different environment. It's not the natural environment, but there's a lot of interactions there, both from treated and untreated human sewage. I'm going to talk a little bit about that in terms of thinking about the design of this kind of work. So, like I said, I think everybody understands the challenge here. And what I want to talk about is really how we're trying to respond to that challenge within NARMS. First, the top bullet there, though, it kind of summarizes, given this complexity, there's been a number of white papers on this topic. And I pull out one of the more recent white papers referenced there, and that link will take you to the report. This was an international effort sponsored by the Wellcome Trust and CDC. The three things that it talks about with respect to environmental waters that I think are very relevant to the conversation is, number one, looking at geospatial distribution, getting better understanding of what the geospatial distribution of resistance. And when we say resistance, it really is all those different elements, the resistant pathogens, the mobile genetic elements carrying those the resistance genes and the genes themselves, but really looking at the geospatial distribution, but not just from a hazard perspective, but really developing that data to inform risk. Second element is really trying to understand the selective, both the sources and the selective pressures for amplification and transmission. Now, clearly there's going to be a lot of targeted, more small scale studies on that, but in terms of the work that we're talking about here, a larger scale national kind of monitoring or surveillance system, the question is, can you develop that system to help get at some of those drivers and sources, questions? And then finally, overarching all this, given the complexity of the situation, the real need to develop standardized procedures was highlighted in this report and elsewhere. And I think that's something that we really want to try to make front and center as part of this pilot, to really develop standard methods for doing this type of environmental monitoring. And then the second box there kind of shows you its verbiage from the NARMS strategic plan, where they really call out, number one, kind of an emphasis on One Health, but secondly, using the surface water as a pilot study. And so in response to that strategic plan, an environmental working group is formed with members from across the relevant agencies, including FDA, CDC, USDA, primarily USDA, ARS, and others, but then also EPA. We kind of gotten involved in this, and I'll mention a little bit about some of the work that we did to position ourselves, or really kind of enter into this field that was relevant to a NARMS kind of program. This pilot effort, first thing we did was kind of define what the objectives were. Obviously, it's a pilot environmental effort within One Health focused NARMS, as you saw from the previous slide in the bullet. But then we also kind of laid out these sub-objectives, targets, really trying to get at more finer scale objectives. One is trying to develop a monitoring plan that could give us a standardized measure to be able to look at long-term changes in resistance in the environment as part of the NARMS effort. And that would include not just the analysis, but potentially a library of samples that you could go back to over time to remine with maybe newer methods as well. And that first bullet really speaks this idea of trying to get geospatial measures of the distribution of the resistance. The second element there is trying to really, though, again, develop data for inputs to risk models. Primarily, since we're targeted on surface water here, it's looking at various risk models, both from recreational use or other various forms of water reuse, either in ag or human purposes. And then the third sub-objective there also is trying to get at this question. While we understand that targeted studies, smaller scale targeted studies on mechanisms is going to do a lot of work or make a lot of progress to get at these mechanistic kind of issues related to that third objective, we also try to design the pilot larger scale monitoring effort to get at some of those as well. And then finally, try to use this information as well to look at critical control points and how to mitigate and improve our mitigation strategy. So also, basically, again, going back to risk and trying to manage that risk. And I should note here too, is that, this is the surface water pilot. We and NARMS is not at all saying that this is the most important environmental element, but it is a good place to start as a pilot for the environmental work. And the reason why we think, that is, why water, right? Why start with water and surface water? As these graphs show, these are all maps that try to kind of give you a sense that, why water? Because there's multiple impacts at a watershed level. And therefore, you really can use the surface water monitoring as a way to really understand those multiple inputs. And really build this, again, the geospatial distribution and risk models in that context of those multiple inputs. The graph on the upper left-hand side, this is a graph it's called the drinks model that was developed to kind of estimate what the overall flow at some of the major drinking water treatment plants in the US, what percentage of the flow is due to treated, in this case, treated wastewater. And you can see that ranges. There's a number of sites that I know the legend's hard to see here. Maybe you can zoom in. It ranges from like 1 up to over 15% of the normal base flow of that surface water where the water is used for drinking water in rivers and mainly larger rivers. That is contributed by just wastewater flow, which may have resistance in it. The graph on the right-hand side in the upper part, that's another human impact of wastewater. In this case, not treated discharges, but the risk from untreated discharges. Those are all of the sites in the US cities that have combined sewer overflow. So that combined sewers, so that when it rains and not overwhelm the treatment plants, the wastewater treatment plants, there is discharge of untreated waste directly into those surface waters. And then the one on the bottom, speaks, again, you guys are certainly experts in this area. I mean, that just is a map there's a EPA tool called the Enviro Atlas, which looks at geospatial information on ecological services and also on ecological stressors. This maps out manure input into different watersheds at the HUC 12 level. So all of these, just to get the message across of why water, because there's these multiple impacts, and we really want to try to understand that as we look to a long-term monitoring program. Now, so I mentioned that we're in the middle of designing the study, and I'll end with a kind of a timeline where we're at in that process. But as you think about designing this study, I think one of the big messages I like to leave you with is this idea of how do you go about that to meet those objectives that I mentioned earlier. I mean, do you want to go big and slow? And when I say slow, I mean not slow in terms of necessarily how long it's going to take, but the frequency of sampling. So do you go at a large scale and sample relatively infrequently so you can get a true geospatial coverage of the US? So the graph that you see, the map on the top of this page, that is from the National Rivers and Streams Assessment that EPA does every 5 years. So we go out and we sample 1,800 sites across the US, and each one of those sites is selected to represent a certain amount of river kilometers. And it's structured around the ecoregions of the US because a lot of this work is focused on looking at the ecological state of that rivers and streams. So really trying to look at, are these waters impaired or not? So it really needs to factor in the distribution and changes in the biotic communities just due to ecoregion. But the point here is those samples are very, when you look at all the dots, it looks like a lot, it's 1,800, but it's really very widely spread. Those are more, but it is designed in a way to be a statistical representation, an estimate of all of the river kilometers in the US. That would be a very valuable tool to get at that objective A of really getting long-term trends and understanding the national scale nature of that. The middle panel there is right below that. Those maps are, we were able to leverage those samplings that were done in this previous cycle and looked at the 50 mil water samples were collected. It's only once per year. So this is base flow conditions in the summer. From those 1,800 sites, we take 50 mil samples. They're brought back to our labs here in Cincinnati, where I work with Office of Research Development. And we do targeted gene analysis to look at fecal indicator bacteria, but we were also able to do some work on some initial screening on some targeted gene analysis of resistance genes, both the INT1 gene as well as SOL1 and the other ones mentioned there. So you get, not to get into details of those maps and what is hot and what's cold, but more that you can use the NERSA design to get at those national maps and look again at long-term trends. So that's kind of big and slow, right? It's 1,800 samples. You got a good picture of the US, but it's only relatively infrequently, once every 5 years. We know that there are other factors that influence the watershed dramatically, like rainfall. So if you want to go small and fast, you want to understand more of the mechanisms, you want to drill down into understanding the temporal variation and look more intensively. The graph that you see there is numbers. This is a CDC study from the Chattahoochee River in the Atlanta area, where they looked at a very highly intensive sampling in that river for both the E. coli and fecal indicator bacteria, but also resistant forms of those. So the big message there is not the details of those slides, but just to kind of get across, go big and slow or go small and fast, or to meet all the objectives that we listed out there, is there a hybrid of those two that we might want to approach? One, a geospatial design that really looks at the long-term trends, and then maybe a standardized approach to look at a watershed level that could be start small, maybe with just several watersheds, and you could build from that, including defining standard procedures and methods, and really a design so you have a consistent approach for looking at it from watershed to watershed. So the next kind of topic is, are you going to design a study where and when you're going to sample, how frequently, but what are you going to analyze? And this page just gets at the need, because of what we already discussed about the genes, the mobile elements, as well as the resistant pathogens themselves, is that you really need to look at the range of them. And the top quote from Amy Pruden from another review article on this topic area of AMR in the environment, given the lack of certainty there right now about what is the best method, you really need to try to look at the range of methods. Certainly from a culture perspective, Enterococcus and E. coli, there's a lot of existing water infrastructure in place to test those things because they are fecal indicators. So trying to build off that and look at resistant forms of those is important. And maybe Salmonella to link to NARMS and the food cycle, but then also look at a targeted gene analysis to really look at the genes themselves, kind of like I mentioned before, as well as the fecal indicators. So you can relate the abundance of the resistance genes to the fecal sources that you see in those rivers. And then finally build in metagenomics to get a deeper look at the overall structure of those communities. And particularly what's interesting these days is that in addition to just doing shotgun metagenomics, but trying to use some of the advanced ligation methods like Hi-C, where you can try to link the genes and the presence of the genes to the taxonomy or the host. So I'm going to finish here with this timeline just to give you a sense of where we're at. We've assembled a team of about 40 scientists from across those agencies that I mentioned earlier. They've been working very diligently over the last couple months to develop really a range of topics, both on designing the study, what's the statistical design, how do you design it so you can end use the data for the models, and then work groups target on all those different analytical targets I mentioned before. They've been meeting. We have a summit planned with that group next week on the 16th and 17th. I guess it's in 2 weeks, where they're all going to come together, going to kind of lay out what their priorities are in those different areas. We're going to assemble that into really the first draft of what we think the pilot study would be. The plan is for that pilot study to occur from 22 through 25. And at the end point there, the real goal is to really inform NARMS on if and what a surface monitoring program looks like for a long-term part of NARMS. And I think what I'd leave you with right now is that, after that summit, really interested in engaging with stakeholders like yourselves to really get feedback and optimize that design as we move forward to the pilot in 22 through 25. So thank you.
All right, thank you, Dr. Garland. Very interesting presentation and a great way to kick off this session that's really kind of focused on this One Health component of environmental and plant health. And so certainly NIAA, as we think about this and my activities at NIAMRI, we very much view this as a One Health type of approach, and often in these types of meetings we don't dive into the environmental or plant health components, and so that's our effort in this session. And so we're going to have a panel at the end of these three speakers. We'll have a panel, so you can go ahead and start sending in your questions you might have for Dr. Garland, and we'll try and get those to him through the panel. And just a comment, many of you probably are aware, but this NARMS strategic plan is out for current public comment. We've still got a couple of weeks left on that, or at least a week left on that public comment. So ask questions if you have them about this aspect, and then consider providing some public comment if you deem that appropriate. So our next speaker is going to continue in this idea of AMR and water and environment, and really look at AMR detection and mitigation in the agricultural environment. So Dr. Michelle Soupir is a professor and associate chair for research and extension in the Department of Ag and Biosystems Engineering here at Iowa State University. Her research program focuses on sustainable water systems with an emphasis on non-point source pollution control, watershed management, and water quality monitoring. Her research projects really are geared to answer basic and applied research questions regarding the occurrence, fate, and transport of pathogens, pathogen indicators, nutrients, and contaminants of emerging environmental concern, which include both antibiotics and antimicrobial resistance to surface and groundwater systems. Her work is focused on the impacts of agricultural practices on water quality and works to design conservation practices to mitigate the impact of ag pollution on downstream waters and reduce public exposure. So with Michelle, we're going to dive into this a little bit deeper and focus in on that agricultural environment. Dr. Soupir, feel free to go ahead.
All right, thank you very much, Paul. And I do not believe I have control of the slide set.
We can hear you. I think they are managing that in the NIAA office, so you can just tell them to advance the slides as you want.
Okay, I think I've got it. So thank you very much for the opportunity to be here today and to share our work related to AMR fate and transport in the agricultural environment. And so to kind of connect this to Dr. Garland's presentation, it's really exciting to hear about the work that EPA is pursuing in this area. This is going to be the small and fast approach, where we're really looking at the agricultural environment and the impacts from manure inputs. And so this first slide is a picture of one of our rainfall simulation experiments where we're testing how these resistant genes and resistant bacteria move along surface flow paths, and then how they can potentially be mitigated by prairie strip systems. So today I'm going to talk through a couple of different, kind of walk you through where it starts to where it ends. So we're going to talk about resistance in manure, what kind of manure treatment strategies could be used, or what kind of mitigation could be used to reduce AMR before it is applied to agricultural fields. Once the manure is applied to agricultural fields, what can we do to try and reduce AMR movement to waters? How can we potentially mitigate AMR at the edge of the field? And then we're going to scale up and look at some agricultural watersheds and some of the transport plans, transport mechanisms that we're seeing at that scale. So why do we care about antimicrobial resistance in the environment? I think that we've already addressed this very well. Antibiotics are used to keep our animals healthy, to either prevent disease or as a disease treatment primarily. We know that a high percentage, up to 3/4 of the antibiotics that are administered to animals orally move through their systems and show up in the manure. So we have manure management systems, manure slurries that have antibiotics, and then also the presence of antibiotic resistant genes. From there, that manure is pumped and often land applied to agricultural fields at agronomic rates. So this is used as an organic source of fertilizer. This example here is showing a manure tanker is actually injecting that manure into the bands. This can occur in the fall or in the spring prior to typically planting corn in the upper Midwest. That water can then flow from tile drains into surface waters and then potentially end up in our water systems. So I do want to talk a little bit about the hydrology in the upper Midwestern United States. So we have a tile drainage system, which means that we would naturally, if you were to come to Iowa 200 years ago, you would see a pretty boggy wetland system. And so tile drains have been installed. So it's similar to a stormwater system in our agricultural fields. This is used to lower the water table, which allows for crop production. We have aerated root systems, and also allows for machinery to have access to fields during those wet periods. But what this has done is it has provided a direct route for the subsurface flow to move into these pipelines and then be discharged into surface waters. So we have kind of bypassed some of that natural attenuation and mitigation that might occur, and moving that water off the field to surface water systems. The areas of and then the picture in the bottom left kind of shows right after a event, you can see, or this is after a new installation of tile lines, you can see kind of what this pattern tile can look like in an agricultural field. A lot of this tiling is concentrated in kind of the upper Midwestern area, which we all know as high areas of agricultural land and crop production. And then Dr. Garland also showed some nice pictures of manure generation. In the bottom right, you can see a map of confined animal feeding operations. So a lot of areas of tile drainage also have a lot of presence of animal production. And so we do need to consider these subsurface pathways when we're looking at environmental monitoring. Okay, so the environmental pathways of AMR have been largely unexplored, and so again, we're going to look at different opportunities for mitigation and how we can try to prevent the movement of AMR into downstream surface waters where we would potentially have exposure for public health. So let's start with the manure management, and this is just talking about a few of the kind of early approaches that we've used to look at different manure management and how we can mitigate AMR. And when I should mention that when we talk about monitoring, we are using both phenotypic and resistant genes. And I can tell you that our results sometimes match up and sometimes they don't. So that kind of speaks to this effort to develop a, either a panel or some sort of a national monitoring strategy that can tell us what kind of indicators we need to use to be able to understand AMR movement through the environment. And so we have looked at anaerobic digestion. I'm presenting here the results of two-stage storage. So this would assume that essentially as the fresh manure comes into a storage system, that manure would then be pumped over to a second manure storage system where it could be held for some period of time before that manure would be land applied. So essentially we would have two manure treatment pits, and the pit that would be land applied would have about 6-month holding time prior to land application. And you can see from the figure on your left that in general, except for that one point at the end, the ERM-B levels, which is a macrolide resistant gene that we have a lot of success tracking in the environment, tends to be pretty stable compared to the fresh manure. So when the fresh manure is continuously added to the system, we continue to see increases in those ERM-B levels. And then another method that has some promising results is centrifugation. And so this is looking at actually the phenotypic resistance, tylosin resistant Enterococcus, and just kind of shows potential spin rates and also time that would need to be treated prior to being able to see significant reductions in the resistant bacteria levels. And so we have been working with Dr. Dan Anderson, he kind of goes by Dr. Manure, on what kind of engineered systems are actually feasible. And we think that both of these have some potential. We do see some potential for anaerobic digestion, especially at higher temperatures. And then there's also some of our collaborators and others working in this field out east have seen some good results with composting. So again, there's some opportunities here to really look at the engineering design of these manure management systems and see what we can track and what kind of mitigation might be possible. So then moving on to the field, so the manure has been applied to the field. What are the trends? What's kind of happening out in the environment and out in the crop fields? And this has been it was a long-term project funded by the National Pork Board, looking at a research farm, one of Iowa State University's research farms, where we have swine manure applied to different times of the year. We have it applied to different tillage treatments. And with our long-term monitoring, we've been able to make a few kind of conclusions about those export patterns. So in general, when we look at the levels of antimicrobial resistance, so resistant genes and also resistant bacteria, we do see that these tend to return to kind of those background levels within about 6 months after manure application. So if manure is applied in the fall, that overwintering we really saw big drastic decreases in those resistance levels. And by spring, those levels in the soil tended to be about or similar to our control levels. We do detect some resistant genes moving into the tile lines, and those detections are higher in the fields that do receive manure. That's not always the case, but over our years of monitoring, we are able to say that we do see some elevated levels of antibiotic resistant genes that are moving into the tile drainage. When we look at spring manure application, we definitely see more export of resistant genes relative to that fall manure application. And another thing that we can say is that a continuous corn system is one where if manure is applied prior to corn planting on a kind of continuous basis, that's where we think there's a little bit more opportunity for buildup of the resistant genes in the soil. So moving on to, okay, so there's just some more to kind of summarize what I had said. Looking at the edge of the field, so prairie strips are a really popular practice that have been shown to have a really effective treatment of surface runoff in increasing infiltration, reducing sediment losses, and so some nutrients that are sorbed to sediment. And so we wanted to see what kind of impact these prairie strips would have on mitigating both antibiotic resistant bacteria and antibiotic resistant genes. And so we conducted rainfall simulation experiments. And so essentially, if you can see that picture in the bottom right, that tarped-in area, there's a nozzle in the middle of that, and it sprays water onto the ground. We have the tarps around it to kind of protect the rainfall from wind. And then we would collect runoff samples. And so this slide gives you a nice schematic of what kind of this experimental setup looks like. But we would apply manure to the small field plots at 200 pounds of nitrogen per acre. So at kind of standard nitrogen application rate for corn. The rainfall would be applied at a rate of 76 millimeters per hour. We often have to pre-wet the plots to get runoff from these. And then we take kind of timed samples from the different outlets. You can see on the right the different layouts that we have. So we have a no manure control cropland with a prairie strip. We have a manured cropland with a prairie strip, and then we have just the manure treatment. And so that can tell us what would come off of a manure treated field, and then what potential there is for mitigation through that vegetated area. We would centrifuge the water samples so that we are analyzing the resistance in the water samples separately from the sediment pellet. The reason we want to do that is because if the prairie strips can mitigate through filtration and settling, we know what could possibly come out versus what might potentially move through the system in the liquid. And also we're doing some pretty intensive modeling of this process. And so modeling what pollutants move through the water flow versus what moves associated with sediment is important for us to know. And so we've used a couple of different technologies. We've used Wafergen, and now we're using Biomark, and this is in Dr. Adina Howe's lab. So I'm an engineer, not a microbiologist, but having a lot of success using the Biomark to look at large suites of antibiotic resistant genes. And so we have postdocs and students that are designing these panels that can really give us a lot of information about what resistant genes are moving through the environment. And so you can see in this figure on the left-hand side all the different genes that we're able to test. And then on the bottom is the sample time. So I mentioned that we collected these water samples throughout the rainfall event. And so plots 1, 2, and 3 are those that were amended with a prairie strip and no manure. And so basically the darker the color means the higher the relative abundance of these different genes. And so you can see that those no manure kind of control strips had our lowest detection of the suite of genes, antibiotic resistant genes. As far as which genes we looked at, we really focus a lot on Sol, and Tet. That's kind of standard as far as what we see in the swine manure that comes from the farms that we work with in this part of Iowa. And the next section is the strip plus the manure. And it's kind of interesting because we had one plot. So each of these were conducted in triplicate. One plot you can see definitely had a much higher detection than plots, that's plot 4, and then plots 5 and 6 were more similar to the plot that was the no manure control. So one thing that we can say that we observed that was different in the field, that higher abundance could be associated because we had a higher flow rate. A little bit difficult to control the flow. Landscape characteristics can have a big impact here, but the hydrology does seem to be very important. And so we could see more export of gene abundance when we had higher flow conditions, even with the vegetative treatment in place. And then the last one that's really a lot of different genes hit up lit up is our manure and plus no prairie strip treatment. Okay, so you can see that there's some potential that the vegetated treatment systems are having some impact on the movement of resistant genes off-site. Another way that we look at this is through 16S gene sequencing. And for this case, we can also show AMR reduction by the prairie strip treatment. And so these NMDS plots are basically used to kind of show similar groupings. And you can see the manure is the dark dots here. The triangles are the sediment, so that pellet that we pulled out, and then the squares are the water. And so first thing you can see is that the sediment and the water tended to, group together. Next, we can look at the differences between the communities from the water samples that received the manure but did not pass through a strip, and they do look more similar to the manure samples. On the other hand, we can look at the communities from the water samples that received manure and did pass through the vegetated strip, and they are more similar to the no manure water samples. So again, this is just another way of looking at this using the 16S data, and we are seeing, that grouping between our samples that were treated with the prairie strip versus those that were not treated with the prairie strip. And so I also want to talk a little bit about watershed monitoring that we've done. I think this will be kind of interesting. Again, we're doing more of the deeper dig where we're collecting samples on a weekly basis, every 2 weeks in some cases. One watershed that we've done quite a bit of work in collaboration with USDA ARS is the South Fork of the Iowa River. And this is just a really high manure application area. Each yellow dot here is the swine system. And what's been interesting here is that in our surface water samples, the percent of samples that are greater than our limit of quantification are up to 80% for ERM-B, almost 90%, and more in the 50% range for ERM-F. But when we looked at the tile waters, we see higher percentages of samples above our limit of quantification. So potentially a little bit more export occurring through tile lines, or there's more mitigation that can take place in the surface water environment. And then finally, I'll talk for just a second about the Blackhawk Lake watershed. This is actually through EPA funding through the National Water Quality Monitoring Initiative. And our point of our study is to really understand if best management practices have an impact on water quality. And so we're monitoring at the catchment scale. This is really small, about 500 acres. And we have a watershed with low levels of management practice implementation relative to high levels of management practice implementation. And what I can tell is that we get a lot of differences from year to year and from site to site. And so when we're looking at tetracycline resistance, in bacteria, so this is the phenotypic resistance, we did see higher export from our low BMP subwatershed in 1 year, but not in the next year. On the other hand, when we look at kind of stacking the different resistant genes that we detected, same story where we see higher export from that low BMP watershed, but the differences occurred in 2018 instead of 2017. So we're not consistently seeing the same patterns from year to year. We can also look at the timing. So do we see resistance that kind of spikes during certain times of the year? I mentioned ERM-B earlier, and again, we can see that during those periods of manure application, which would be probably post-harvest and then probably into pre-planting, we did see a little bit of increase in tylosin resistant genes, but we weren't able to detect those same trends for ERM-F or TET-M, which are two other genes that we have a lot of success monitoring. So I want to wrap up here and just, emphasize that there's a lot of different areas where mitigation can occur. We can see elevated levels of resistant bacteria and genes, but the resistant bacteria and genes do not seem to really correlate with each other. So there's lots of remaining questions. I'm really excited to see how Dr. Garland and the work done with EPA moves forward, especially when we think about risks to human health. So we're doing all this monitoring in the environment, but But what does that do for human health? And so then I definitely want to acknowledge a lot of different funding sources that we've had over the years: National Pork Board, EPA 319 through Iowa DNR, and then several grants from USDA NEFA and NSF, National Research Traineeship Program. And this is clearly a work in collaboration with USDA, ARS, and other folks at Iowa State. So these are some references, and I will turn this back over to you, Paul.
All right. Thank you, Dr. Soupir. Very interesting presentation. I think we can see a lot of opportunities there where, you know, we can start to evaluate some relatively practical ways that moving forward, we can better understand how we can mitigate and improve our best management practices and alter those to help, you know, better understand the role of agriculture, animal agriculture, and impacting environmental levels of antimicrobial resistance and bacteria. So I'm sure we'll have some questions here when we get to the panel. But for right now, we're going to go to our third panelist. And this may seem like a little bit of a shift, but again, all falling into this kind of environmental impact and environmental components of antimicrobials and the One Health associated with plant health. So our next speaker here is Dr. Christopher Vincent. And Dr. Vincent is an assistant professor in horticultural sciences at the University of Florida. His research program is focused on serving Florida perennial fruit crop producers with production methods and biological understanding to improve productivity of citrus crops and additional crops and cropping systems in the traditional citrus region. In particular, he studies how plants move water and carbohydrates within themselves and in response to environmental factors. And in the particular case of antimicrobial compounds that we're going to talk about today, along with collaborators, he's been assessing the degree to which antimicrobial products enter and circulate within citrus tree to reduce the bacterium that causes citrus greening. So I suspect he'll give a little bit of background, but you may be aware that there have been some approvals for use of antimicrobials to control this citrus greening in orchards. And so he's here to provide us a little bit of an update on that and what they're learning through that process, with the hopes that we can understand that and think about how that impacts our approaches to manage other One Health components of AMR. So Dr. Vincent.
Awesome. Well, thank you very much. Thank you so much for the opportunity to speak. I'm glad to see that there's interest in this topic. Again, my name is Christopher Vincent. I'm an environmental physiologist studying mostly citrus at the University of Florida. And the title of my talk is Where It's At: Do agricultural antimicrobial compounds reach pathogenic bacteria inside the plant? And of course, I've been very clever and put a little target on the portion of the plant that we're trying to reach. And I'll explain a little bit about kind of what that means and why that's important as we go along. Just to give a quick overview of what I'll be talking about. First, I'll introduce Florida citrus, the context of citrus greening, and the use of antimicrobial compounds in the fight against greening. Then we'll take a look at some studies of efficacy of antimicrobials against citrus greening. And then we'll talk about delivery and systemic movement of those compounds as well. And then we'll finish up with foliar assessments of foliar application in terms of delivery. And we'll shortly get to exactly what I mean by delivery. But first, I want to start with the context of citrus greening. You'll notice in this picture on the left-hand side, that's not a very healthy looking tree. It's not healthy looking because it's not healthy. It's infected with a bacteria called Candidatus Liberibacter asiaticus. And that bacterium inhabits only the phloem of the plant. Now the phloem is the vascular tissue that transports sugar throughout the plant. So what that leads to is dysfunction of the phloem or a lesser ability of the plant to move sugars from the leaves to the roots and to the fruits, and that inhibits just overall growth, not to mention yield. Some of those symptoms are like thin canopies, like you see on the left-hand side, and also this blotchy mottle symptomology that you see in the bottom right here, compared to a nice dark green, healthy citrus leaf that's not infected with the bacterium. Now, plants can continue to grow and produce after having been infected, and that's the situation for the majority of Florida citrus. Most plants in the field are now infected and have seen reduced growth and yield as a result. However, production is still occurring, and we'll get into kind of how that's happened and what the overall effects have been in just a second. Somehow I'm having trouble moving my slides now. There we go. Oh, too many. I apologize. I think it's moving forward. Can we go back to that slide that I was at and then move forward one? I apologize. It doesn't seem to be responding. I think we need to go back one more slide. There we are. Oh, sorry, one more still. Here we go. Okay, so this is a plot of total citrus production in the United States from 1998-99 through 2017-2018. The reason that you have groups of 2 years is that the season, actually the harvest season, stretches from roughly November through April, and so April of the following year. So we look at 98-99 as being 1 year and 99-2000 as being another. But what you can see is that at the beginning of this period, Florida citrus provided the majority of the total citrus production in the United States, and you have a general decline across this period. Now in 2004 and 2005, there were catastrophic freezes that dramatically impacted production in those years, as well as for a few years afterwards. However, at that same time was the first time that the Liberobacter bacterium, which was already known to cause greening, was first discovered here in Florida. And since then, it has spread throughout the state in approximately the subsequent 5 years. And then you see that just sort of long, slow decline of Florida citrus from about 2008 through through today. And that decline of citrus has been both a decline in yield per acre and a loss of acreage. So as growers are forced to increase their inputs in attempts to manage the disease, while they face declining yields, it's pushed many of the smaller growers out of the citrus business. And so we've fallen from about 900,000 acres of citrus in Florida to now slightly over 300,000 acres since 2004 through today. So you can see that with that decline, why citrus growers would be desperate for measures that would stem that decline and possibly help improve production, help them stay in business, essentially. So in 2016, we had a Section 18 emergency exemption approved for 3 products. 2 of them are streptomycin-based products, and 1 is an oxytetracycline product. They were all previously used in apples and pears to manage fire blight. So their use in trees had already been demonstrated, and it was effective in that system. And so it seemed reasonable to go for use of antimicrobials as a stopgap measure to manage huanglongbing or citrus greening. So the the long-term goal for citrus growers is to develop resistant or at least tolerant varieties and strong production methods. However, this rapid decline and loss of growers and acreage put people on the hunt for sort of rapid stopgap measures that would stave off the decline until those other long-term methods that are slower to develop could be implemented. Next slide, please. Okay, so starting in 2016, we have the use of oxytetracycline and streptomycin in Florida citrus. You'll notice that the oxytetracycline data that we have only goes 2016 and 2017. And the reason for that is that in 2017, a permanent label usage of oxytetracycline was approved. And so we don't have Section 18 emergency exception based data collection. So we don't have the data for 2018 through 2020. However, what you see is a general decline in the use of streptomycin, and we would probably tend to assume that the oxytetracycline follows a similar pattern. And I have a question I want to, okay. Just, yes, to be clear, the production cycle is traditionally was about 30 years to 50 years. But the time from planting until bearing for modern varieties is roughly to maximum or full production is about 5 years to just lower level commercial production is about 3 years. So from planting until production, you have 3 to 5 years. So you can see why long-term solutions take a really long time to develop. In 2016, we had pretty widespread usage. The application of both types of products together is estimated to have been made on about 330,000 acres. So that's a significant portion of the existing acreage at the time. And it had an economic impact as well. So on the right-hand side, you see these are just pie charts of the cost of applications made to citrus foliage. And you see in 2015-2016 season, which is prior to the approval of bactericides, of course, you have no cost there, and then for bactericides, but then in 2016 through 2017, all of a sudden, growers are spending on average $50 or almost $51 per acre on bactericides in particular. In that same survey, our economist, Dr. Ariel Singerman, determined that growers were reporting that they had reduced expenditures in nutritional, so that's foliar applications of mineral nutrients, and insecticides. Those insecticides were primarily being used to manage the vector of this disease. So they reduced costs in nutritionals and insecticides in order to be able to afford the cost of the bactericides. However, that's the year in which we believe that the greatest amount of bactericides were applied, and you see a general trend of decline in the subsequent years, and so the most recent survey indicates that growers spent on average about $10 per acre on bactericides, and that's not so much that most growers are spending, how should I say it, that the growers who are using bactericides are spending less, but rather that fewer growers are purchasing and applying bactericides. Next slide, please. So beginning just prior to the approval of the Section 18 exemption, the Citrus Research and Development Foundation, which is a grower-directed, state and grower-funded research agency, They began large-scale grower trials, replicated trials in growers' fields, and beginning in 2018, significant body of results began to come from those, and the results were very ambiguous. So some growers saw statistically significant increases in production that they believed were a result of bactericides, and other growers saw statistically significant decreases. There were discrepancies in analysis. Various groups used the data and provided their own analyses with differing results. However, one of the claims that came from the very public discussions about the efficacy of these products was that adjuvants in particular would have a big impact on the efficacy of these products to fight this bacterium, this Liberibacter. So you see on the right-hand side, an article from the New York Times, spraying antibiotics to fight citrus scourge doesn't help, study finds. So that was a study that came from a microbiology lab group at our research center. They were comparing trunk injection to spraying these bactericides on the foliage. And they found essentially that they couldn't detect delivery when oxytetracycline was applied to the foliage, to which the company that was marketing these products responded that, oh, they used the wrong adjuvant. So just to be clear, an adjuvant is a product that you mix in with other products in order to enhance either their penetration or their adherence or some physical property like that would affect its efficacy. So in this case, the adjuvants that we're talking about are penetrant adjuvants, so they should enhance the penetration of the product into the leaf. So that's essentially, these types of questions are exactly how I came to be involved. I'm not a microbiologist. I don't study bacteria. However, what I do study is how things, or stuff, as I like to call it, move around the plants. So usually we study sugar and water. However, we also have the tools to investigate the movement of, say, oxytetracycline and streptomycin. So our questions were essentially, do oxytetracycline and streptomycin enter the plants through the leaves? And also, do they move after having gone into the leaves into the phloem in particular? The reason why this is important is that because the bacterium exists only in that particular part of the plant, these compounds can only be successful in managing the disease if they are in fact delivered into the phloem. When I say delivery, what I mean is the journey of a molecule, oxytetracycline or streptomycin, to its intended destination. In this case, this would be the phloem of the plant. So what is its path? So there are a few ways that we're going to stick with oxytetracycline for the moment. There are a few ways that oxytet can make it to the phloem. So in this micrograph, you see the purple outlined cells, those are phloem cells. And a plant can come through the stomata, which you see in the top left-hand side, and move through sort of in between the cells and eventually make it to the phloem. It can also penetrate the cuticle, which is a waxy layer on the surface of the leaf, and make it through either through the cells or in between the cells to the same location. If it can make it into the xylem, which is the vasculature that transports water, it can also be delivered through the inside of the vascular bundle to the phloem. So when we talk about delivery, let me just mention in particular about citrus. I apologize, the slides are responding, but I'm being impatient. Citrus in particular has a very thick waxy cuticle that prevents the escape or the entry of water. And it's very difficult for compounds to make it through the stomata because the stomata are actually equipped with these wings of the cuticle that come up on either side and prevent that when the stomata are closed, they actually are doubly closed because the waxy cuticle also closes over those stomata. So it's very difficult for water to get out of the leaves. And likewise, it's very difficult for water bearing oxytetracycline, say, to get into the leaves. So to address this question of whether oxytetracycline might make it through the phloem, my colleague Nabil Khalini labeled some oxytetracycline with green fluorescent protein. And so this is that GFP labeled assessment. And so what he did is he just applied that oxytetracycline with a fluorescent dye to the bark of the tree and then watched where it went, essentially by taking cross sections. What they discovered is that most of it moves in through the xylem, which is what would be expected because the xylem being the portion that moves water, it just moves the most volume. And they did find, however, that after moving into the xylem and moving throughout the plant in the xylem, that it does cross from the xylem into the phloem. So in terms of whether oxytetracycline can make it into the phloem, well, we have pretty strong evidence that yes, it can, and it probably will. Then they applied non-labeled streptomycin and oxytetracycline to roots to see how both of these compounds move throughout the plant and how easily they were moved. And essentially what they found was that streptomycin did not move very easily. When it was applied to the roots, most of it stayed in the roots, some of it moved into the xylem, some moved into the phloem, and very little made it all the way up to the leaves. Streptomycin, on the other hand, did move quite readily, mostly into the xylem and then some into the phloem and then a little bit less into the leaves. They also did an additional experiment where they girdled the tree, when essentially what this does is remove the phloem in a particular portion. And they found that that didn't actually change the amount of oxytetracycline that made it into the phloem. So essentially that phloem delivery is dependent on xylem movement and not on delivery directly to the phloem. So it's actually a good sign in terms of if you're thinking that if your goal is to deliver oxytetracycline to the phloem. And so this is that study that found that foliar application didn't help very much. And essentially what they found was that when they applied oxytetracycline to the leaves along with an approved adjuvant in the field, they had no detection of oxytetracycline. They then moved that into the greenhouse which is a more protected environment, and they found that after a few days, very small amounts of oxytetracycline were detected, but that they also didn't last very long. So by 14 days, the concentrations were already again below the detection limit, and not anywhere near the concentrations estimated to be needed to actually reduce Liberibacter. So because of the ongoing controversy about efficacy and the role of adjuvants in particular in affecting results, we went ahead with a field study, and we just decided to use all of the approved adjuvants and compare them to find out which ones were effective and which ones could be recommended to growers to use in applying oxytetracycline. So in this case, we used 6-year-old trees. They were infected with Liberibacter. We applied about 1.56 grams per tree, and we used all of the approved methods as well as injection, and we had a negative control as well, which was water without oxytetracycline, water with oxytetracycline. This is essentially what that looks like in the field. In the top right, you see Ronnie from my lab installing a tree injector. On the bottom left, you see Mirta from the lab tying a bag around the leaves. And essentially what we're doing there is, by bagging off some of the leaves, we are protecting them from the direct application. And so any oxytetracycline detect in those leaves has been moved in from outside. And that's essentially the part that we're looking for. We're not quite as interested in how much oxytetracycline is in the leaf because we know that it can be trapped in various places that aren't related to phloem delivery. So what did we find? On the right-hand side, you see the total results. You see that injection resulted, we're talking about same total quantity of application per tree. Injection resulted in more than 5 micrograms per gram fresh weight delivery of oxytetracycline in the leaves. And all of the other treatments had much lower concentrations. And while there is variation in the means, you can look on the left-hand side and see this is kind of a blown up view of that same plot that's on the right-hand side. There are variations in the means. However, the amount of variability that we find there, essentially, there are no significant differences there between any of the adjuvants and the negative control. What I mean by that, the negative control, I mean water without oxytetracycline. Excuse me. So that's the one on those are the controls are the two on the far left hand side. You see that when you apply oxytetracycline with the water, you don't actually find more oxytetracycline than you do when you don't apply any oxytet at all. So essentially what we're seeing here is that we're playing with our minimum limits of detection. And none of the adjuvants led to statistically significant increases in detectable oxytetracycline in the leaves. And by statistically significant increases, I mean increases over not applying any oxytetracycline at all. The place where we did find differences was between uncovered and covered leaves. So the leaves that did receive direct application had higher concentrations, but because what we're interested in is systemic delivery, it's covered leaves that are our indicator of delivery that will affect Libraribacter. And so we see that demonstrated in this plot here. This is a qPCR-based detection of Libraribacter populations. So in this case, actually, a higher cycle threshold means a lower bacterial population. And so what we see is, over time, the only treatment that led to a decrease in the bacterial population or the increase in the CT value is this injection treatment. All of the others remained essentially the same over time. So in terms of conclusions, what we see is that the impacts of the use of oxytetracycline in the field depend on delivery to the phloem. And what we see is that essentially systemic movement does occur, and oxytetracycline is moved around the plant, though streptomycin seems to be much less mobile. However, in terms of actually achieving delivery, very little of these products that we're applying to citrus foliage appear to be making it into systemic movement. And that challenge with delivery, I believe, actually explains the lackluster effects that in our field trials and has led to a lot of this controversy. And so with that, I'd like to thank everyone who works in my lab, as well as funding from the Citrus Research and Development Foundation. And thank you very much for your time.
All right. Thank you, Dr. Vincent. And very interesting. Certainly a unique perspective that we often don't have. So look forward to questions and discussion here. So I believe we're going to transition back to our panel format now. And so we still have some time for questions here, and we have a nice question period built in. And so we'll pull up Dr. Garland and Dr. Soupir and Dr. Vincent, and we can probably get rid of the slides here. And so a couple of questions have already come in, and we'll go through those. You started answering one of those, Dr. Vincent, and so just for continuity, I'll finish out that question. And so that was, how are the bacteria transmitted between the trees? Is it in the soil and taken up into the roots, or is it from tree to tree by mechanical transmission, or how does that occur?
Yeah, so the Liberibacter is transmitted between citrus trees by an insect vector, and that's the Asian citrus psyllid. And as far as we know, there's no transmission, mechanical transmission. It can be transmitted by grafting if you're using infected budwood, but that's not an issue in the citrus industry because we have a well-regulated nursery practices.
All right, excellent. So now a question for Dr. Garland, and maybe also some follow-up for Dr. Soupir if she has comments. So when AMR bacteria are found in the watershed, in the NARMS plans specifically, do you intend to investigate further to determine the source of that bacteria? And if so, how would you do that? And would you be considering all sources, kind of the human, animal, wildlife type of potential sources?
Yeah, and it's a very good question. I think it partly gets back to the question of what type of design you're looking at, and we may be looking at a continuum of both. But if you talk about the big and slow, right? So the large scale national surveillance, I think two things. We certainly are interested in looking at all of the sources. The way to look at that, there's twofold. One is to look at the metadata about the watershed that you are sampling there, and understanding not necessarily specific sources, but types of sources within that overall watershed. Second thing is that we hope to build in, I mentioned it briefly, about looking at fecal source tracking markers into that panel of targets so that you can look at the relationship between certain genes and other measures of resistance and the source of the dominant sources of fecal contamination in that surface water. And also by looking at that relationship, you might be able to see, where do you see higher levels of resistance genes than predicted from any given fecal source tracker, suggesting that those might be conditions leading to the amplification of the genes, not just the attenuation of the original fecal sources, but potentially amplification. So that's a very good question. Really, we really want to build that into the design, but probably more broadly, right, from the watershed's perspective, not from specific targets within the watershed, if that makes sense.
So how do you address the wildlife component there? I mean, you probably don't have good, background samples for source tracking of the wildlife. So,
increasingly though, there is a lot of good targets for various types of wildlife, particularly different kinds of birds and also other mammals. So that is an area that's, I think, kind of rich for analysis, not only for the metagenomics, but we do a lot of that work within the EPA because we're obviously very concerned about fecal source tracking, because from risk models, you really want the human fecal source is really going to drive risk more than other fecal sources. So we really have looked at trying to distinguish from general fecal indicators to more specific ones. And so I think that's where we really want to build that, and I know Dr. Soupir talked about this too, about a panel, this high throughput fluidine biomark system where you can really look at a large number of targets and try to rapidly assess. And I think one of the keys is trying to define a standard panel that includes this very, kind of standard set of and a wide range of fecal source trackers in addition to ARGs.
Excellent. Dr. Soupir, any comments? Obviously, you're doing more specific application, and so you're applying, but I didn't know if you had any comments to add to that.
Yeah, I mean, I agree with what Dr. Garland said, about integrating the source trackers into this panel. I think that'll be really helpful, and it's quite frankly what our stakeholders want to see too. That's one of the #1 questions we get when we're working with farmer groups is, well, where's the bacteria coming from, right? And so if we can figure out how to answer that question, it really helps with designing mitigation efforts as well. I would add that from the perspective, one thing that we have done in our, very kind of digging in when we have a known manure source is we look at like a soil column study, and we put the manure on and we rain through and we collect what comes out the bottom of it, then we can identify that manure associated community, so we can kind of identify the genes, and even at a slightly higher level, what are the microorganisms that are derived from that manure, and we can pull that out from the kind of environmental background data sets. So we've been able to do that with a lot of geese samples. We worked with Iowa Department of Natural Resources to do that. We've done that with swine manure and beef manure. So a little bit different than kind of the standard source tracking, which is often looking at like bacteroides, but definitely, it would be great if we could figure out where stuff is coming from in the process.
All right, great, thank you. Another question, Dr. Garland, and this is fairly specific, so this relates to the NARM strategic plan and the inclusion of this water sampling there. And that relates to, obviously NARMS has historically struggled to be well funded. And so as you look at, as the strategic plan looks at potentially expanding the scope of that work, do you anticipate that this water sampling through EPA would be pulled out of that normal NARMS budget, or is there a separate budget for that? And what implications might that have more broadly on the NARMS program, which obviously is of interest to folks at NIAA?
Understood, and it's a very good question, one that comes up when we have that's come up in these working groups. When the scientists get together, it's like, well, how much money we got, right? How much resource do we have to do this? And we've really tried to focus them and the entire effort right now on the science first. Let's define the priorities. What is the minimally acceptable scientific design? And so the summit that I mentioned that's coming up in a couple of weeks, we've asked each one of those working groups to say, this is the minimally acceptable design, or this is the minimally acceptable targets, and then here might be more optimal approaches. And we really want to kind of rank out things from a priority perspective. So that's number one, really focus first on what is what has to happen from a science perspective. Secondly, we really are working to try to leverage resources across the agencies. I mentioned that NERSA program. I mean, that's something that exists and on that 5-year sampling, so we could leverage off of that. So and that includes both the extramural funding, but also the internal scientists within EPA, USDA, ARS, and their time and commitment to the effort. So leveraging is important. And then finally, on specific budget issues, this kind of, like a zero-sum game, where's it coming from? I really, not to be dodgy, but I would really leave that to maybe Pat McDermott with FDA, who runs NARMS, kind of ask that question more directly to him. And I think he would agree that we're really trying to work on developing the plan first, and then really defining how we think it's the best way to fund that, and really trying to leverage resources wherever possible.
All right, excellent. Dr. Vincent, a couple of questions just came in for you, and some commendation for a very interesting presentation. And one of the questions was, why was treatment of citrus trees the chosen approach? Was the cilia control too hard, or why was that approach the approach that was taken with the emergency exemption and stuff?
Yeah, so the growers had spent the previous 10 years focusing on vector management, right? And although vector management was very successful in reducing psyllid populations, the cost of an infection is very high, right? So it only takes one psyllid to infect one tree, and insecticides typically have their impact after the psyllid has already arrived. And so you may kill that vector, but it may have already succeeded in vectoring the disease. And this is a persistent pathogen. So once it's infected, unless you can, inject it with enough oxytetracycline or something, it's infected for life. So over those previous 10 years, essentially what we had was the spread of this disease through every single planting in the state. And when I say every single planting, I mean, like 90% of the trees being infected. So essentially what they were looking for was something that would help them reduce the disease in trees that already had it. So that was why they went in that direction. They think that vector management helped slow the disease, but it was already progressed so far that at that point it was, they felt sort of time for chemotherapy.
Yeah, sounds good. Second question was, and so, obviously in animals, when we use antibiotics, we have what's called a withdrawal period, and, we can't market product from those animals until a particular period. The question is there any withdrawal period for antibiotic application to citrus? And how have you seen consumers respond? Obviously we've spent the last couple of days talking about messaging in the animal health side. What's your experience on the plant side been, and the consumer response and the withdrawal?
Yeah, so the equivalent in plants to a withdrawal period is the pre-harvest interval. So essentially what that means is that the grower can't apply the product within a certain number of days of harvest. And I don't know off the top of my head exactly what that pre-harvest interval is. I want to say it's like 2 months, but I could be wrong on that. It may be a little bit more. So that does exist. I don't know on what basis it was determined, because I've never seen strong evidence. Well, there's only one study that I've seen strong evidence about how much oxytetracycline or streptomycin move into a citrus fruit. However, there has been a lot of consumer concern about antibiotics in these products. We're giving orange juice to our kids in the morning, and they're drinking streptomycin and oxytetracycline, and so far I don't know what evidence there is that that's something that they should be concerned about, in the sense that the concerns make sense. However, based on our data, not very much of these products are making it into the plant at all. So it's really at this point more of a concern for why should growers apply it in the first place. However, for instance, if growers decided to go with like injection, and that would require additional approvals, because at this point only foliar application is on-label use. If they decided to go with injection, then there would be more concerns about that sort of thing, because then we know that it is moved in the plant, it does make it to the phloem. And the reason I mentioned phloem in this particular regard is because that's how things get into the fruit, is they're moved through the phloem into the fruit. So at that point, those concerns would then probably come to the fore again. There have been a number of articles in the Washington Post, the New York Times. There was a national story on Good Morning America. Lots of parents concerned about what they're feeding their children and that sort of thing. And I think that in terms of how the industry and researchers responded, it was very much a game of, it was a reactive game rather than a proactive game, where these sorts of concerns were the kinds of things, this is just my personal opinion, of course, but they're the kinds of concerns that you could have anticipated. Mm-hmm. And so they could have perhaps put some information out there to begin with that would sort of, allay the concerns of consumers. But what ended up happening is, it's these expose type stories that are being done, and it makes them look bad. It made them look bad.
Sounds great. Appreciate that. Was a great response. So going back a little bit to the probably Michelle, I'm guessing for the first shot at this question, and then others can respond. Has there been work to look at the persistence of AMR in lagoons at animal operations and what sort of factors influence their presence. I think you commented a little bit in one of your slides on that, but maybe expound on that and talk about it a little bit more.
Yeah, so I can say that when we look at the manure, we see high levels of resistance, and we do typically detect antibiotics in these lagoons. And so the challenge is if we study the lagoon with time, we're typically having new inputs of manure coming in on a daily basis. And so a lot of the work that we have done has been in the lab to try and isolate and control for some of these environmental factors. And so in that, when we separate out the manure, so that if we pull the manure and then just let it sit, we know that there are decreases in levels of antimicrobial resistant genes. So by separating out that system and from the fresh inputs that are coming in a daily basis, just time alone will have kind of exponential decay, and we see that impact as far as being able to decrease the levels of AMR. You know, there's a lot of interest in anaerobic digestion. So can we, you know, use manure to produce gas and have some warmer temperatures that could also kill off resistant genes and pathogens in the process? I can say that we haven't had a lot of success with this in the lab. It almost seemed like we were preferentially selecting for some of the organisms that carried those resistant genes, and we saw some increases. So, you know, that's just some lab-based work, but there has been some work on composting. So if we can separate out the solids and compost, there have been others that have reported good decay of resistant genes in a composting system. And one other thing that we actually have a new project starting on this, but looking at the manure from a lot of different systems and the impact of the veterinary feed directive. So the recommendation to only use antibiotics, not for growth promotion, antibiotics of concern for human health, not for growth promotion, but for, you know, disease treatment or prevention. And also, you know, the consumer demands for antibiotic-free meat. And so we'll be collecting manures from lagoons that come from these antibiotic-free systems, and also from you know, the more traditional treatment systems, and looking to see what kind of differences we see using these gene panels like the biomark that we've been using in our environmental samples. And because the fact is that we think the persistence can kind of linger even if the system does not use antibiotics. We honestly, in our kind of preliminary studies, are still seeing that resistance. It could be from other feed additives that are not necessarily antibiotics, but maybe they still can have some of that same impact. So I think there's a lot of hope that with the VFD we'll see a decrease in resistance, and really hope that some of our kind of future work can help to support that.
Great, appreciate that answer. Still got about 5 minutes here, so another one I'll throw out is related to, so it kind of ties into Dr. Vincent's comments, but I'm interested in the environmental side. You know, that both Dr. Garland and Dr. Soupir, you talked about AMR and AMR bacteria. I think you briefly mentioned, though, the antibiotic concentrations, right? The actual antibiotic themselves. And so just some perspective and comment on that. And then for Dr. Garland, you know, has that ever been part of that NARMS discussion, or is there any intention to looking at that as well?
I can start on this. We have done a fair amount of monitoring for antibiotics through Tom Moorman's lab, and now Diana Aga at University of Buffalo is doing a lot of this work with us. And I can say that when we go out in the environment, it's really hard to get detects of the antibiotics. So if we're in a stream system, we put these POSIS samplers out. They're like little sorbent materials, and we go and pull them every 2 weeks or once a month, and then we can kind of back calculate a concentration based on time and flow if we have that information. And so really takes kind of some different sampling techniques to be able to detect the antibiotics in the environment. And then I guess it's also important to look at the metabolites. So there's we can try to detect the antibiotics themselves, but how are they breaking down in the environment? And we think that some of these these metabolites may even be more important in the role of development of resistance and possibly amplification than the initial target antibiotics. So, but I can tell you that work is complicated. It takes a lot of time. It's hard to extract the antibiotics out of these environmental media. And so there's just, kind of a handful of labs that have really got good methods developed for this.
Yeah, and I guess I would add that, I mean, I concur that it's difficult to measure. So sometimes the attractiveness of the fecal source trackers as a way to look at sources as opposed to the, you could look for the antimicrobials themselves and try to relate that to potential where it's being used in humans, say, versus agricultural sources. But it's a little bit more difficult to look at those as opposed to the gene targets, especially with some of the the qPCR techniques. You can have very, very low detection limits. So, but the discussion has come up about using those as markers too, in addition to the molecular markers.
All right. Another question here as we finish up for Dr. Vincent, related to the potential species of trees that would be susceptible to blight. So you obviously talked about being citrus. Are there non-citrus trees that are susceptible to the same organism, or maybe a quick overview of where else antimicrobials or antibiotics might be used in tree production or something?
So this bacterium only infects trees that are closely related to citrus. So we have a couple of other species that are used as ornamentals here in Florida that can be infected by the bacterium, but it's pretty much just citrus limited. And as far as other trees where antibiotics may be used, they are used in apple production in places where fire blight is a concern as well, and apple and pears are similar in that regard. Off the top of my head, I'm not familiar with any other tree in which bacterial antibiotics are used.
Excellent. All right. And then a question and a comment that came in. From Dr. Soupir's work, it sounds like much of what you've found, and correct if this is wrong, that you've learned about AMR and movement of that mirrors what we found in the Midwest related to nitrate flow. More water, more rainfall equates to more flow of nitrates, and in such cases, AMR and bacteria into the system. So if that's true, Dr. Garland, would the small and fast approach make more sense to the development of the NARMS approach and provide a design that yields more actionable information for the stakeholders?
That's a really good question. I think I tried to emphasize the ideal situation would be to have a blend or a hybrid of those two approaches. One to look maybe at long-term trends and maybe almost baseline conditions, but certainly, like I said, the rainfall events, these perturbations to the watershed, in essence, those may increase the risk level. So you really want to capture, in essence, the worst case scenario, understand what the boundaries are of the level. So I really think where we're trying to move towards is, as I mentioned a little bit, is maybe there's a way forward where you have the long-term big and slow study to look at these longer term trends, but then really develop a standardized approach to look at a watershed and then try to pilot that as a way then to replicate it across multiple watersheds into the future. Right. So the pilot might be really focus a lot on defining the standard operating procedure basically for doing that watershed study, which would account for trying to capture the temporal effects within the watershed and other mechanisms that are going on, and then build off of that to try to expand it out in the out years, even beyond the pilot, right? To really think about how you would, again, leveraging other programs, even through NSF and folks that have these existing like NEON, which is an ecological observing network? How could you build into these other programs to have a small and fast, but replicated small and fast across multiple watersheds in across the nation?
All right. Well, I think we're about out of time. So I want to quickly kind of summarize and, really appreciate all three of you coming to speak today. They were very interesting topics. We started off with Dr. Garland giving us an update on the EPA and NARM's water sampling approach, and providing some context and insight into how that timeline and development process is going, and answering some questions that our audience had related to that. And obviously NARMS is really important to our country's strategy around the National Action Plan, and better understanding what role EPA and that project may play as the strategic plan for NARMS plays out. Then Dr. Soupir stepped in and talked to us about these antimicrobial resistant organisms and resistance markers in agricultural environments, something very specifically around animal agriculture, and something very specific and directly applicable to a lot of those of us in the audience, and provided some insights, as she mentioned here a few minutes ago, in terms of lagoon and mitigation there, but also talked about some differences in times of application of manure, and those certainly would be things that we could approach and manage through best management practices. The contribution of continuous corn on corn for those of us in the Midwest that understand the economics of corn production and stuff. That's a significant impact, and so how that impacts our application processes and stuff. And then some really interesting work on those prairie strips and the potential for a very ecologically useful prairie strip to help us mitigate these resistance markers, and certainly an approach that we want to further understand. And then bringing up on the end with Dr. Vincent with a topic that was very unique and new, I think, to most of us in the Zoom discussion, that know zero about orange trees, in my case, other than I love to drink orange juice, but that's about where the extent of it ends. And so better understanding the use of antimicrobials there, certainly as somebody involved in the AMR and One Health, I was aware of that, but the level of understanding and the scope, actually, maxing out at, I think you said 330,000 acres of land applied, product, significantly down from that now. But I think you also laid out a model there where you had producers that were really wanting to use that product, but when they really got into the science of it, they started to understand it maybe wasn't doing exactly what they thought it was doing, and that there was probably other ways that they could better use those products to improve efficacy and minimize. And you also highlight the significant impact that antimicrobial cost has, because any of these uses plays back into the economics of our production cycles, and so that's just kind of another example of that One Health. It's all tied together in our environment, with our livestock in it, our plants, and humans as well. So appreciate, again, all of you for participating in this panel. I think it's been informative. I think from the number of questions we had and the diversity of those questions, the audience was clearly interested. And again, thank you for your time. And so now I would like to turn it over to Dr. Michael Costan. Dr. Costan, you saw yesterday with me as well. I think Eric said earlier it was the Paul Plummer show. It's probably equally the Mike Costan show. And so Dr. Costan served on a panel yesterday. He works as associate director at AVMA and provides staffing for the AVMA, American Veterinary Medical Association Committee on Antimicrobials. His background is as a dairy practitioner. And so he's got a clinical background and now works obviously with AVMA in the area of policy and and antimicrobial use. And so Dr. Costan's going to be our moderator for our next session here, getting some updates on the National Action Plan and national other national issues, probably more specifically, that relate to the federal government. So Dr. Costan.
Thank you, Dr. Plummer. I hope everyone has enjoyed the symposium so far. I think we've heard from some amazing speakers who've shared a lot of new and interesting information, as Dr. Plummer said, that I was not aware of before. So thank you to our previous panelists. In our next session, we will hear from several representatives of the U.S. government about the international landscape and the process related to antimicrobial resistance, and learn how the U.S. interacts within that landscape. At the end of this session, we hope you have a better understanding of the current levels of the U.S. government's interaction at the international level, and how documents produced at the international level impact the United States. It is now my honor to introduce our three panelists. Dr. Nina Anandaraman currently serves as Veterinary Science Policy Advisor to the United States Department of Agriculture's Chief Scientist. She focuses on antimicrobial resistance, biorisk, and zoonosis. In coordinating antimicrobial resistance activities across USDA and with other federal partners, Dr. Anandaraman co-chairs the Task Force on Combating Antibiotic Resistance Bacteria on behalf of USDA. She is also a member of the Transatlantic Task Force on Antimicrobial Resistance and serves as alternate U.S. delegate to the Codex Resistance. Our second panelist is Dr. Clara David, Biodefense, where she covers antimicrobial resistance and global health security issues. She joined the office in January 2020 after serving as the Science and Technology Advisor to USAID Indonesia, managing programs in Indonesia to build capacity in science and health. Prior to joining USAID, she worked at the National Institutes of Health. Our third panelist is Dr. Lynn Philpe. Dr. Philpe currently serves as Senior Global Health Advisor and Antimicrobial Resistance Team Lead to the United States Department of Health and Human Services, Office of Global Affairs, and the Office of the Secretary. She is a member of the Federal Task Force for Combating Antibiotic Resistance Bacteria and the Transatlantic Task Force on Antimicrobial Resistance. In her current role, she leads in conjunction with USDA and state the International AMR Working Group, with the CARB Task Force sets international AMR policy. Dr. Philpe represents the U.S. government and Department of Health and Human Services as a member of the Global Health Security AMR Action Package, the Global AMR R&D Hub, and the Presidential Advisory Council on Combating Antibiotic Resistant Bacteria. There will be a question and answer session at the end of the panel discussion. If you have questions, please enter them in the Q&A box, and our panelists will try to answer them at the end of the session. Doctors, thank you very much for joining us today. And I believe Dr. Anandaraman, would you please start us off?
Thank you so much for that introduction. And I just, real pleasure to be here. And I just want to let you know that my presentation is the first part of really a three-part presentation. And I'm going to kind of start out with the history of what brought us to where we are today. And then my colleague at the State Department, Dr. Clara Davis, is going to go more into detail on the different entities, international entities that I'll very briefly mention. And finally, Dr. Philpe is going to talk about where we are currently and also look to the future. Thanks. Next slide. So just to give you a foundation for those that aren't aware of it, we have the Federal Combating Antibiotic Resistant Bacteria, or CARB Task Force, and that's co-chaired by HHS, USDA, and the Department of Defense. And it has, as you can see here, a number of participating agencies and departments and offices. And I mention this because the work that we're doing on the international coordination it really falls under the auspices of this CARB task force under the CARB National Action Plan Goal 5. And you may wonder how so many different agencies and offices with very different missions and visions are able to come to agreement on anything, much less the whole global community coming to agreement on anything. And really what sets the basis for interactions is science and risk. And I'm going to be coming back to that point further in the presentation. Thanks, next slide. So with this timeline that kind of sets us up for what's going on today, I'm going to start from left to right, and all of the activities that are above the annual year boxes are all domestic activities, and what's below it are the corresponding international activities. And so starting off at the top left, you see the CDC Threat Report, which was first released in 2013 and updated last year. And that threat report, again, sets that scientific basis for the AMR public health issues that guide our work. And that same year, you have the National Security Council convene an interagency policy committee, which is the internal US government experts, and asks them to develop a national strategy to address AMR. And then you have the President's Council of Advisors on Science and Technology that serves as the external group of advisors directed to come out with recommendations. And then looking below 2013, you see what was then the G8 makes AMR a priority issue for the G8 countries to address, which then provides that political will and resources to address this issue amongst G8 countries. Then looking at 2014 at the top, you have an executive order released that year that has three major directives. One is a CARB task force is stood up by HHS, USDA, and DoD and directed to take the national strategy that that interagency policy committee came up with and the recommendations from PCAST to come out with a 5-year CARB National Action Plan. The second item in the directive order is HHS Secretary with advisement with USDA and DoD is directed to stand up a Presidential Advisory Council for CARB, and that serves as the external group of experts. And then the third directive is international coordination is asked to be conducted by HHS's Office of Global Affairs, State Department, and USDA, hence the three departments that are speaking to you on the panel today. And then in 2015, the CARB Task Force releases its CARB National Action Plan, and then the White House holds a stewardship forum, both on the human side and the animal side for stakeholders to provide that momentum to implement this CARB National Action Plan. And then below, you see on the international side, the WHO releases its Global Action Plan, directing all countries to develop their own national action plans. Then you have the Global Health Security Agenda, or GHSA, starts implementing its action packages, one of which is antimicrobial resistance. And again, you'll hear about these in the other presentations in more detail. That year, you also have the FAO and OIE come up with resolutions endorsing what the WHO's put out in its Global Action Plan and resolving to help address from their scope of work. And then finally, you have the TATFAR, the Transatlantic Task Force on Antimicrobial Resistance, releases its 5-year action plan. It was actually begun back in 2009 on the human health side, and in 2014, an animal component was added. So then all these years, the G7 is continuing to include AMR as a priority item. And that's moved to the G20 in 2016, and then the G77. And that political momentum is then moved to the UN General Assembly in 2016 for commitment from global leaders across the world to address AMR. And the FAO and OIE also released their action plans and strategies that year to help implement the work of the WHO Global Action Plan. And then you have the UN General Assembly directing the tripartite WHO, FAO, and OIE, actually the member states of the General Assembly directing that tripartite to work within their sphere of expertise to work in a multi-sectoral way to approach this issue. And this was just the fourth time that a health issue has ever been brought before the UN General Assembly. It forms an external group body, the Interagency Coordinating Group, to develop recommendations in the next couple of years, and you'll hear more about that in the next presentations. And then finally, in 2017, the G7 chief veterinary officers come out with definitions around some gray areas of therapeutic use, prevention, control, treatment, and others. And you have a Codex task force on antimicrobial resistance that stood up again to update a 2005 code of practice and to develop guidelines for antimicrobial resistance surveillance. And again, all of these are being covered in more detail in the next couple of presentations. Next slide, please. Thanks. So on this one, so I just do want to cover 3 of the international organizations, and that's Codex that has a food safety mandate, the OIE that has an animal health mandate, and actually it's International Plant Protection Council, not council, it's convention, and the IPPC is a plant health mandate. So these 3 organizations are known as the 3 sisters under the SPS. They're also known as the international standard setting bodies. So they have this dual role of protecting health, but also ensuring fair practices in food trade. And so why these 3 organizations? Next slide, please. Here you'll see Article 5.1 of the SPS agreement. It says that measures must be based on risk. And at the bottom of that, you see that you have to take into account risk assessment techniques developed by the relevant international organizations. And those relevant international organizations are those 3 sisters or the international standard setting bodies, because their very basis is science and risk. And if a country wants to take a measure that's not represented there, they need to provide scientific justification. So next slide, please. And so I just want to leave you with the USDA Science Blueprint that was just released earlier this year. And it has 5 themes: sustainable ag intensification and green up top, and then the food and nutrition translation at the bottom there specifically call out antimicrobial resistance objectives. And then I want to spend a little bit of time on the ag science policy leadership, one that's up on the left, that addresses a lot of issues, but a particular objective I want to read out that's pertinent here is to advocate globally for the development of science-based international and domestic standards, regulatory approaches, and policies, including those guiding the development of new and emerging technologies. And so you can follow implementation of this blueprint at USDA Science and other topics at that Twitter feed if you like in the future. And with that, I'll turn it over to Dr. Clara Davis. Thanks.
Great, thank you, Nina. So what I'd like to talk about a little bit next, following up on Nina's explanation of the process over time, is a little more of the specific landscape and the various international organizations and the various documents, because this can be kind of an alphabet soup with all of these different groups. And health diplomacy, which is really these international organizations and the international interactions dealing with health-related issues, is a whole of government approach in the United States. The State Department is in charge of the relationship with other countries and with multilateral organizations, but we work closely with the other domestic agencies, both as part of the technical expertise and links with the technical agencies in other countries, but also, very importantly, to look at the domestic effects of international actions. So one of the first ones on the timeline was the G7, the Group of Seven. This is a leaders level summit. It was begun, and I'm sorry, I just realized the slides are not going forward. JJ, could you go? So sorry about that. Thank you. The Group of Seven is a leaders level summit begun in the 70s as a forum on economic issues, though this is now expanded to include health education and other global issues. At the start, this included approximately 80% of the world's GDP, but now the Group of Seven covers approximately 50% of the world's GDP. Next slide, please. The G20 was formed in 1999 to expand the discussions at the G7 to include additional rising economies. This now includes approximately 80% of the global GDP. It includes all of the G7 members plus 13 additional. Both of these groups do meet yearly. The duties to host shift among the different members because these are not formal organizations with headquarters. They provide opportunities to discuss shared issues, release statements of shared opinion, but they don't negotiate treaties or other forms of international law. And if we could move to the next slide, please. The other big G group is the Group of 77. The United States is not part of this organization. The G77 was formed in 1964 as a coalition of developing economies. It's now expanded to 134. As you can see, there is some overlap with the G20, but this acts as an organization to focus on shared action primarily in the UN setting. Next slide, please. The Organization for Economic Cooperation and Development was originally formed in 1961. All 37 members are high-income countries that are dedicated to democracy and the market economy. This is a forum to discuss economic issues, compare experiences, and coordinate the policies of members. It publishes non-binding recommendations of principles, standards for business conduct, and it also provides a major source of expert reports, reference materials on a range of economic issues. Next slide, please. I'd like to shift to one body that Nina mentioned that is purely health focused. That is the Global Health Security Agenda, which was launched in 2014. It now counts over 70 countries and organizations as members. Again, this is a forum. It brings together countries and organizations to share experiences and best practices focused on the spread of infectious diseases globally. It does not create binding laws. It is there to promote communication and collaboration, not treaties or fund research. It also does not actually directly do outbreak response. It's about our shared global interest in preparing for outbreaks. And next slide. Again, I wanted to point out that at the formation of the Global Health Security Agenda, there were 11 action packages. Antimicrobial resistance is one of these focused action packages. So it is a focused effort at the GHSA. And this has now been expanded to 12 action packages. Next slide, please. Now I wanted to shift to two of the major organizations relevant for creation of international law, the Treaties and Conventions. The United Nations and its subsidiary organizations can create binding treaties, but this includes organizations like UNICEF, World Bank. The ones we're most likely to think about in an antimicrobial resistance context are the World Health Organization and the Food and Agriculture Organization. In many cases, they do not actually create specific law. The World Trade Organization was also created as a treaty, so it is a form of international law that was established to create a transparent system to encourage trade among countries. And next slide, please. I wanted to bring up the Codex Elementarius, which was also mentioned on Nina's timeline. This is a body that creates the shared food standards and guidelines for international trade. Importantly, while it creates the standards and guidelines, it's up to each country domestically to implement those standards. But this is important because the World Trade Organization refers to these standards and officially uses them. So the World Organization for Animal Health, the OIE, also is one of the standard setting organizations for the World Trade Organization. And next slide, please. There's kind of a hierarchy for these different documents that you might see referred to. Treaties or conventions are actual documents of international law. Countries which have ratified them are held to these terms. Regulations are generally a step below, but they are a form of law to be followed by states that have ratified them. A major example in our context is the International Health Regulations. This was agreed to by the international community, the current version in 2005, and it is about how to deal with infectious disease outbreaks. Now, as you go down, guidelines and standards are relevant for agreed upon definitions, resolutions and statements, which are a great deal of what you see. Many of the documents on that timeline are resolutions or statements. These are more often a shared opinion of that body that are not typically considered a form of law, but instead are opinion and are generally reached by a consensus process. And each member country during a resolution can still issue an objection to pieces of it. This would be termed an explanation of policy or an explanation of vote, which sort of carves out that item which that specific country has an objection to, even if it agrees with the general goal of the resolution. Recommendations are just that, a suggestion to member countries or others. And expert panel reports, they're an important method to collect evidence and can feed into all the other kinds of documents, but they're not a decision in and of themselves. And next slide, please. This is one small example of one of the documents. The Global AMR Action Plan called for every country to have a national action plan on AMR, which we'll talk a little bit more about in the next talk, and also was mentioned yesterday. The US and many other countries responded. The US does have a national action plan, and we work on this as a whole of government approach. And part of that plan is that we do collect information on our progress. We voluntarily report back to the international community on that progress. And the goal is that this creates a sort of feedback on this. We all report, we share our experiences, share the lessons learned, and translate them into our own domestic settings, and each country can adjust their work as needed. And next slide, please. So what I'm hoping you come away from this with is not just that there's an awful lot of organizations dealing with this topic at the global level and how complex some of these negotiations are, but that this is not just about negotiating a formal treaty. Part of this process is about coming together, bringing different parties to the table and creating a consensus about need for action or types of actions to take. And by building that consensus over time, you keep the whole group moving together towards that joint action. It's also important to come to agreement on shared definitions, a common language, so that we all agree on what we are saying, and to be able to use science and risk as the basis for that common language. One other element, and this has especially been a part of some actions like the GHSA, is shared action below the levels of treaties or laws. For instance, shared action could involve agreeing to the importance of an issue, cost sharing, actions we all agree to, or creating a new avenue of interaction. That might mean meetings of experts, educational exchanges or programs, joint capacity building, or humanitarian response programs. And hopefully, this has given you a little bit of an introduction, and I'd like to hand it over to Dr. Lynn Philpe, who's going to talk about the next stages.
Thanks, Clara. So Nina and Clara gave you a very nice overview of what's been happening globally over the last 5 or 6 years, and what all of these international bodies we work in mean and what is and isn't legally binding. I'm going to try to speak a little bit more to what the US has done within that landscape, and hopefully make it relevant to this particular audience. But if I don't, please do ping us in the questions and ask a bit more. So that is not advancing, but next slide, please. So as Nina mentioned in her timeline, in 2015, we not only came out with a national action plan, but the WHO came out with a global action plan. And these things were closely linked. A lot of the same experts who were working on creating our national action plan were also advising the WHO on what theirs should look like. And since that point in time, we have actually seen some major progress made. A few highlights are the CDC has led in creating an AR lab network, which takes a regional approach to surveillance domestically. BARDA and NIH have worked together to start CARB-X, which is an accelerator for new antibiotics, non-traditional therapeutics, and diagnostics. And while that was called for in our first national action plan, it very quickly transitioned into a global initiative with partners from the UK and Germany, Wellcome Trust, and others, and is hosted out of Boston University. We also pursued alternatives for antibiotics in agriculture and human health. And again, led by the CDC, we obtained hundreds of commitments to global action in the 2018 AMR challenge. That was a UN initiative where the US sponsored side meetings and called on all global partners to take a year to think about what more they could do. But while the task force and domestically we were achieving all these things, the international landscape was undergoing quite a shift. The major goal of work in 2015 and 2016 was just to gain political will. AMR was largely seen as a problem of donor countries, and specifically a very small subset of donor countries that had already identified it as a high human health burden, because those were the countries that were looking for it. Those were the ones seeing it in human health. And those were the countries that were vocally calling attention to AMR as a global issue and saying, just because you haven't found it yet, doesn't mean it's not there and it's not affecting your human and animal health. This particularly focused on the gram-negative bacteria because there were already global programs to address things such as TB, HIV, malaria, etc. And there was a group of health ministers at the WHO called the Alliance of Champions, and the US is a member. And those health ministers got together and really started pushing for political will. They did it by pushing the WHO to do more. They did it by rallying all the countries to really push for the tripartite to refocus on AMR. The tripartite, the work of WHO, FAO, and OIE does work on other things such as influenza, but hadn't really focused together on AMR. And that was a problem because this is a One Health issue and we need the experts from human health, animal health, and food safety all together in the room leading on this issue. We also pushed for this to be a priority topic at the G7 and the G20. And as I already mentioned, the US has helped in obtaining hundreds of commitments globally for people to do more on AMR. But one of the notable things that happened was the push for the high-level meeting at the UN. Next slide, please. So as Clara and Nina both mentioned, when this conversation moved to the UN, it pulled in the G77. And pulling in the G77 was pretty critical because it was no longer just an issue that donor countries were focusing on. We now had leaders from all countries looking at this and thinking about how it was relevant to their countries. And really that caused a huge influx in countries that actually developed national action plans, and more importantly, were willing to dedicate some resources to those national action plans. This resulted in a declaration. That declaration called for a lot of coordinated action, but specifically it called for a UN interagency coordination group on AMR, and this became the IACG. I think many folks are familiar with the IACG by now. This was a group of expert advisors from in and out of the UN system. It was not necessarily government representatives, although there were government representatives participating, but they were doing so in their personal capacity as an expert. And this group came out with several reports and recommendations for the global community on how we can come together and do more on AMR. And then in April of 2019, they came out with this report, which I hope many of you have seen, No Time to Wait, Securing the Future from Drug-Resistant Infections. And this was a report delivered to the Secretary General of the UN with very thorough, very detailed recommendations on what everyone should do on this issue. These are good recommendations. The US position on this is that we simply cannot implement them all. Some of them are not the responsibility of member states. Some of them fall to non-government partners. Some of them fall to the international organizations themselves. Even with the US budget to combat AMR, we can't do everything on the list that we want to. And so we do have to prioritize. And you can see that through our national action plans. But these are recommendations. They are not legally binding. They are meant to offer advice and a framework for future action. And they're meant to offer that to countries at all levels of development and economic income. They did make, though, some recommendations on government structures to the Secretary General, and the Secretary General, in response to this report, directed the tripartite with implementing those governance structures. So I want to talk a little bit more about these structures because I think they do affect people on this call. They are One Health in nature, and they are meant to deal with human health, animal health, and food safety. So the first is an independent panel, or sorry, the first is a global leaders group. And this one is the one that's the farthest along. This is meant to be a very small group. We're not quite sure on size yet. WHO is going to announce this group during World Antibiotic Awareness Week this year, which happens from the 18th to the 24th. This is really meant to be high political level advocates for AMR that can push for this to be maintained at the UN, the G7, the G20, et cetera, that can really push that political will. Then there is the independent panel on evidence for action against AMR. This group is not very far along yet. The tripartite has not asked for public comment yet on their proposals for how this group will function, but they will be shortly, so please watch that space. And that panel is really meant to be a small panel of about 10 to 15 experts that will really look at the scientific evidence and make recommendations on why we should take this action in the first place. The last one is the partnership platform, and I think that's the one that will be of most interest to those on at this meeting. The partnership platform is meant to facilitate a multi-stakeholder engagement in order to develop a shared global vision on how to approach AMR. It's supposed to be a venue for information sharing. It's not quite clear what that means yet, because this is also one that is still being created. But it is meant to be the largest group of the three. And the tripartite has indicated this one expects membership in the hundreds. They really, they're not limiting who participates in the partnership platform. This one is meant to bring in all those with an interest in AMR and want to be part of this conversation and moving these efforts forward. And the last thing I'll mention is that along with these three global governance structures, the tripartite has asked the UN to set up a trust fund, an AMR multi-partner trust fund. And this trust fund is specifically meant to implement a work plan developed by the tripartite to assist lower middle income countries in building capacity. There is still a lot of conversation on how these four bodies will work together, not duplicate work, but work separately and work well together. So that is also ongoing. And again, other than the global leaders group, all will be coming to the public for comment in the next year. So please, again, watch that space. So next slide, please. So Nina had mentioned the strategy that was developed back in 2014, 2015. And this came out of the work not only of PCAST, but of the CARB National Task Force. It was directed by an executive order that was re-upped again by this administration. And so it can be summarized, excuse me, it can be summarized in infection prevention and control and stewardship. Goal 2 is often surveillance. Goal 3 is diagnostics and treatments. And, goal 3 is diagnostics, 4 is treatments. And goal 5 is all of the things that we cover, all the equities that the US government has in goals 1 through 4, but taking it internationally, making sure that we are advancing the equities that need to be advanced and that we are protecting the equities that need to be protected. So specifically on the international side, we are to improve international collaboration and capacities. Next slide, please. And as I mentioned before, we are seeing progress. And this is from the current CDC Threat Report. And in recent years, overall deaths from resistant infections have fallen. Specifically, deaths from resistant infections in hospitals have gone down. We know that more US hospitals are implementing high quality antibiotic stewardship programs. These programs are resource and time intensive. And this has been a concerted and a heavy lift. Next slide, please. But there is still a lot to be done. We, through the threat report, are still estimating that there are 2.8 million antibiotic resistant infections each year and over 35,000 deaths. So there are too many resistant infections, too many deaths still. We also know that new resistant pathogens are emerging. Candida aureus was not included in the 2013 CDC Threat Report, but is now considered a serious threat. And CDC has reported that some resistant infections, particularly those acquired in communities as opposed to hospitals, are increasing. There's also significant ongoing challenges to developing new antibiotic products. And not just new antibiotics, but alternative therapies, vaccines, diagnostics, etc. Next slide, please. So, as mentioned, we just released the next 5-year action plan. This will last from 2020 to 2025. In putting together the second action plan, we started with an assessment of what we had done and where the major gaps still remained. We asked PACCARB, the Presidential Advisory Council on Combating Antibiotic-Resistant Bacteria, for their recommendations. And we also looked at the recommendations of the international community, including the IACG recommendations and the individual advice from the international organizations. We also went back to our bilateral meetings and looked for specific asks from our global partners. Next slide, please. And the next steps that were pointed out to us for goal 5 were pretty consistent. Keep doing what we're doing, keep working on capacity building with other countries, keep improving surveillance and stewardship and infection prevention and control. Keep up our work on research and keep up our work on economic incentives for new antibiotics. But there was an overwhelming message that the US wasn't doing enough in a leadership role. This kind of surprised us, to be honest, because we felt that we were showing a leadership role. We had led negotiations at the WHO on an AMR resolution. We were a member of the Alliance of Champions and numerous other fora where we put our political will. We were very vocal in support of the actions happening at the G7 and G20. We were very involved in AMR coming to the UN. And so what we realized was we really needed to do more on making our international work public, and we needed to do it on a level where it had a political policy impact. Next slide, please. And so again, one of the things to watch in the coming year is that in order to sort of solve that problem, we have proposed that we appoint a US federal champion for international CARB. And this would be a person of a high enough level that they could speak on the international stage and advance the US policy in this area. But this position doesn't currently exist. And so we have to create it. And over the next year, OGA, my office, Nina and Clara's offices as well, will be working with the CARB task force to put together an options paper for the secretaries of HHS, USDA, and state to consider appointing a champion. And this will need to look at where this person will sit, what this person's responsibilities will be, will there be funding attached, et cetera. There's lots of questions out here. And the reason why I point this out to this group is that this is also a space to watch. At some point within this process, and it's just begun, so I don't have a timeline for you yet, we will be asking for public comment. And we do value the comments of our stakeholders and what can the US do better to promote US policy and to push ourselves and the globe into more action to really make a dent in antimicrobial resistance. So next slide, please. And I'm not going to go into everything we're going to be doing in Goal 5 because that would probably take up the entire hour, but just a few more things that we're doing that might interest this group. We're going to, of course, continue our work with international partners, and that includes the G7 and G20. We are, as of tomorrow, taking on the Global Health Security Agenda's AMR Action Package chair, and we are going to continue to actively engage with TATFAR and get that work plan out. So I'm going to talk a little bit more about those fora. Next slide, please. Nina went into this a little bit, but just to provide a little bit more detail. The G7 is really where the political will, international attention on AMR began. In 2015, Germany held the presidency and they pushed for the G7 to produce a best practices brochure and called for the UN high-level meeting. That brochure is a little dated now, but there's still some good information in there. And it's probably something the G7 will take on again or would push to the G20. The next year, we discussed the international review on AMR. Some of you may remember this was a report developed by Jim O'Neill's group out of the UK. It was really one of the first arguments, economic arguments for why this action on AMR should move forward. And then as Nina mentioned, under Italy's leadership in 2017, we developed some One Health definitions. What we found was that in these political documents, there is confusion with the terms we're using. For example, the US uses control, the EU often uses metaphylaxis. What do we consider therapeutic? What do we not consider therapeutic, et cetera? And so there was a One Health working group as part of the agriculture track that really looked at that. Those definitions are still out there and they are still agreed upon by the G7. But what was pretty clear to the G7 was that we needed more global partners for this to really make a dent. And so there was a big push to move it to the G20. The G20 is usually focused on economic issues. And so it was a little bit of an argument at first for why the G20 should take this on. But they have, and they've gotten some real commitments out of it. So in 2016, the agricultural ministers committed to phase out medically important antibiotics for growth promotion. And this was then welcomed by the health ministers. The G20 has consistently called for economic incentives for new drugs, but also to help us maintain our old drugs, to solve some of the global manufacturing and access issues. And in order to help do that, in 2017, we called for the development of a global R&D hub. So next slide, please. And that hub has gone live. It's hosted by Germany. And it's still in early days, so it's still collecting a lot of the data that's out there, but it's working with all global partners to do so. And the US is a member of the board for this hub. So we do often provide them with feedback on where their work is going, but this hub is not to be interfered with politically by member states. We guide the work, we tell them what information we need, and then we leave the experts to do the work and to provide us the information that they're gathering. They're focusing on three main areas right now: investment to AMR R&D in general, antibacterials in clinical development, and incentives for antibacterial R&D. And one of the things that I'd like to point out is, again, this is still very early days, and they haven't come out with many analyses yet, but they've already found through reaching out through the G20, but additional partners as well, that on the animal side, there are really only 73 funders for AMR R&D on the animal side. There's roughly $560 million US spent, and it's probably about 768 projects that they've been able to track. When you compare this to the human health side, there are 86 funders on the human health side. There is 2.48 billion USD spent on the human health side, and over 1,500 projects that are being worked on. And this is a little bit of, of need. This is a high human health burden, and so a lot of funding does get spent to it. But I think this also highlights for us how much more support we're offering to the human health side than we are to the animal health side. And that is something that's already been identified and is under discussion by the G20 and the country members of the Global AMR R&D Hub. The hub also does have a stakeholders group where they reach out to non-government partners as well. So again, I welcome everyone to go, if you Google Global AMR R&D Hub, it will take you to their site and the dashboard, and you'll be able to see all the information that they have been collecting. A lot of it is still to come, and they tell you where that is as well, but feel free to go on there and look around. Next slide, please. So one of the other places where we do One Health work, and this is more on a technical level, is the Transatlantic Task Force on AMR. TATFAR was created in 2009, so it predates the CARB group. And if you look closely enough, we go back even farther in the US on concern for this issue and how many programs we worked on. But TATFAR was one of the first formal fora that we engaged in with international partners. TATFAR's technical experts collaborate with our international partners, and TATFAR membership right now is the European Union, Canada, and Norway. We are in the middle of working on our next implementation period. The priorities of improving antibiotic use in humans and animals, preventing infections and their spread, and strengthening the drug pipeline will remain. Those were the original priorities of TATFAR. But in doing an assessment of where we are now versus where we were in 2009, we're looking to refocus ourselves on what the current priorities should be, where that technical communication should be happening. This allows us to better understand our partners as we're negotiating these global governance issues, but it also really helps on things like integrated surveillance on the global stage. If we can talk to the EU and Canada and Norway about our differences, then we can all come to the table with a mutual understanding of how to go forward. And then the last fora, next slide please, that I'm going to just briefly talk about is the Global Health Security Agenda AMR Action Package. As I mentioned, we're taking over the chair as of tomorrow morning. The GHSA Ministerial is going on right now. It's hosted by Thailand and it is virtual. The talks, unfortunately, are starting about 5 a.m. or earlier in the East Coast time here in the US. But there are some really interesting talks on health security preparedness, especially in light of the current COVID-19 pandemic. And in fact, tomorrow morning at 5 a.m., there is a side meeting on AMR in the time of COVID that we are co-hosting, and then will be the transition between Uganda's chair and our US chair. So why those at this meeting might care about this. So we've already mentioned this to you a little bit, Global Health Security Agenda. When this was originally created, it was really meant to help countries look at their ability to respond to the international health regulations. And to do that, we set up these joint external evaluations where countries are asked to do a self-assessment, and then a team of experts comes in and looks at their self-assessment and also does an assessment of their own, and countries are given a score. They're then asked to put together a strategy to improve that score and look for action to increase their capacity building. And so under our chairmanship, we'd like to refocus on really helping countries look at their abilities on AMR and their abilities to respond to the international health regulations and their scores on the JEE, and think about how they can improve and where they need to improve. And then they can come to the donor countries and ask for partnerships and help. And frankly, the donor countries can also do our assessment and look for the places where we also need to improve, because there's certainly a lot there as well. Also, the tripartite led by the WHO is putting together a benchmarks library that will specifically provide tools, recommendations, guidances, etc., to all the GHSA members, so that when they identify an area where they need to improve their capacity building or their JEE score, they can go in there and search for advice on how best to do that. And so under our chairmanship, we'll really be looking for ways to help strengthen that library and do that assessment. And membership in GHSA is not just countries. It is also non-government stakeholders. So this includes the international organizations, but it also includes financial institutions, regional organizations, etc. So it is open to all who want to focus on being better prepared for global health. And I will end it there. There are more fora, please feel free to ask us about it and open it up for any questions.
Not here. Thank you. Can you hear me?
Yes.
Good. Thank you very much, Dr. Zanandaraman, Dr. Davis, and Dr. Philpe. We don't have a lot of time, but we have received a couple of questions. I'm not sure which of you can answer this one, but can the panelists discuss enforcement process when a member country chooses to not follow the agreed treaty?
So, yeah, Clara, do you want to take that?
Well, I mean, I can give the very general answer, which is that it depends on the treaty. As with any other kind of rule, it depends on if there's enforcement mechanisms built into the treaty, and also kind of then the actions that the other signatories would take as a result. So I don't think there's a single answer. Nina, do you want to add?
No, that's probably, yeah, that's great. I really don't have a good response for that. Thanks.
And just to add, we actually don't have any treaties or legally binding agreements on AMR. The closest thing we would have would be the international health regulations, and they don't actually call out AMR, although it does fit in other ways. And so when Global Health Security Agenda was being created, that's why AMR has a dedicated action package, so that we could kind of bring that to the conversation there. But still, the work of the action package, as Claire already mentioned, is not legally binding. The recommendations and the commitments in the G7 and G20 are the responsibility of the countries to take them on. The G7 does do an assessment where they talk about if we've fulfilled the commitments we've made in the previous years. So we do get publicly called on the carpet if we don't in the G7, but the G20 doesn't have that mechanism. So again, it depends on which one we're talking about, but we don't have a treaty on AMR.
Thank you. Another question, this is for Dr. Davis. How do we help lower income countries create an antimicrobial resistant national action plan that are based on science? I guess the questioner thinks that, there's some examples out there where rather than implementing infrastructure that would help, blanket bans of antimicrobials have been done. Is there a way the U.S. is helping in this role?
So I'll take it as a broad question on helping on AMR, not just on the national action plans, which we certainly can assist with. And I think that gets to broader questions about how to do a kind of effective capacity building and international development in this area. The key is that when we go in and we work with other countries and we offer our assistance, we work closely with them to design how we're going to assist them. And then this is something where different parts of the US government might work with them. USAID has had programs that are relevant to antimicrobial resistance. Also, the CDC has had global programs working with other countries. And as we go in and we present our expertise and assist with our technical knowledge in helping shape that, then that is the primary way we're going to look at both what the science shows in terms of what's going to be effective, but also linking it together with other areas that are going to be effective in AMR, such as basic sanitation and hygiene questions and such. So I think the key element to your answer is, as we go in and offer assistance, that we make our assistance based on the science and based on the knowledge of what will work. And then we also work together with other countries that are also offering assistance.
Thank you.
I think we have time. Can I just add to that before we move on real quick? Because specifically on AMR, we did have the global action plan from the WHO, and that was meant to provide a framework. But the reality is this question is showcasing, is that a lot of countries basically copied it and then put it on a shelf and really weren't able to implement it. And so, as Clara mentioned, USAID and CDC both specifically have bilateral relationships where they help countries build this. Some of that work was done through the Global Health Security Agenda. Some of it is just through the ongoing work of those two organizations. And then, often when I mentioned earlier that we went back and we looked at our notes from our bilateral meetings, what I meant by that is that when our leaders are at these large public meetings, they have bilateral chats with their counterparts. So, for example, Secretary Azar would be talking to health ministers from other countries. And we are often point blank asked for assistance on AMR. And that those asks are taken seriously and are sent down to the working level so that we can put our experts in touch with their experts and do the best we can to help guide them. We, of course, have to take into account their budget and their abilities and where they're at in their capacity building, because some countries we're talking about, they just need to improve their surveillance a little bit. And in other countries we're talking about, they don't even have hospital beds. So, so there's not a one size fits all approach, but CDC and USAID both have excellent bilateral programs. And then as part of CARB 2.0 that we've just released, CDC is also looking to take more of a regional approach, and we'll be doing that as well to hopefully set up some abilities regionally, resources allowing, that can help countries not just work bilaterally with us, but work together among themselves as well to sort of share best practices and work on how to actually implement these plans.
Thank you. I appreciate all three of you taking the time, and on behalf of NIA, thank you very much. After this session, I think we all have a better understanding of the international arena and how the U.S. government interacts in that forum. I'm now going to turn this over to Dr. Andy King, who will provide a wrap-up as it reflects to communicating about antibiotic stewardship and antimicrobial resistance. Dr. King, the floor is yours.
Thank you, Dr. Costan. And so, yeah, my job here is to sort of wrap everything up and then turn it over to Leah and Eric for their closing remarks. And so just a reminder to start things off. Tomorrow, you will get an email with a post-symposium survey, and I know that's come up already, but please, when you can, complete that survey. I know that surveys go out for all sorts of things and all sorts of conferences, and email at this time of year is overwhelming, but please, if you have a minute, please fill out that survey and provide us with feedback on the symposium across the board, just the content that was covered across all of the different areas. And then obviously, personally, I'd be interested in your feedback on the communications component as well. And that's particularly relevant, I would say, because in January 2021, that's 2 months from now, there will be a communications quarterly meeting based in part on the feedback from the survey that I just mentioned. And so the quarterly meeting, as we're sort of thinking about it, is aimed at providing attendees with opportunities to discuss how they've applied the knowledge, insights, and ideas from this year's and last year's symposium about communicating with audiences of interest, which could even be potentially internal audiences at your company, agency, coalition, or organization. So one important aspect of that survey that we're, again, hopefully you'll see in your inbox tomorrow, and that you'll take the survey. It'll be there, just I hope you'll take it. So beyond providing feedback about the program overall is how can NIAA help you in your organization solve specific communication problems that you face. And so that was the goal of this year's communication session compared to last year's session, which is getting us from sort of those general communication principles and encouraging you to tell stories and thinking about sort of what are best practices, into sort of remembering sort of past missteps, some current approaches that really work well, and some of the things that are happening in agricultural communications training to hopefully sort of feed employees to you eventually that'll help you communicate your stories in time. And so the goal of these sessions is to dive even deeper and try to be more solution oriented with problems that you face. And that'll help us develop a program moving forward as well, thinking about communications at Symposia in the future. You're welcome to contact JJ and the folks at NIAA directly about participating in that January session, and I encourage you to do so. I'm sure I'll be involved some way, and so I do really hope to hear from you and hope that we can continue to improve our communications programming for NIAA. And with that in mind, I do sort of want to sort of end my closing wrap-up thoughts with the challenge to folks here, and that's to pursue communications opportunities that are relevant to you as the calendar year closes out, and to try to improve your social and digital media presence for your operation, if that makes sense. And so part of the continuing communications efforts of NIAA include providing a toolkit to help you share your stories via social and digital media outlets. Again, if you're interested in that, I encourage you directly to reach out to JJ and NIAA. And that's the purpose of that, is to try to give you more concrete steps moving forward to help you share your story, the story of your company, and the story of your operation. And so with that, to close things out for the entire symposium, I will turn things over to Leah and Eric to wrap things up and provide their closing remarks. And again, I hope to hear about your thoughts on the communications efforts through that survey, which goes out tomorrow.
Great. Well, thank you, everyone. I think, Leah, we've closed another symposium, and if everyone indulge us for just a few minutes, we would like to provide some closing remarks and then give you a challenge at the end that Leah is going to throw out there for you. But I think one thing that just amazes me is the quality of people that are willing to participate in the conversation. I want to do a big thank you to all our speakers and give you my impression of how the 3 days went. And so we just heard Dr. King talk about communications, and I'll mention a few speakers, but by all means, all speakers did a great job, and we truly appreciate your participation, as well as we appreciate the participation of the audience out there as well, and everyone's on here, knowing that this isn't the end of the discussion. We need to carry it forward. So if I could recap the last 3 days, and we started communication with Dr. King, you know, it's all based on One Health. Truthfully, the environment, humans, and animal health. And if we break that even further, it's building off the individual up through the environment, up through to a global level. So day one really was focusing on you, the individual. How do you communicate? What do you what's the important things to express your values? We build that base that builds all the way up to day three when we talk about policy, global policy and global action plans. So day one really was building the dialogue. And so hopefully you can take those tools away, take the challenges that are given to you to move forward and have those tough conversations. And so that was one leg, the human side, the one person in that One Health thing. If we look at the three-legged stool of One Health, on day two, we covered two of the legs of that stool, animal health and human health. And I think if you saw the progress and the activities around monitoring antimicrobial use and resistance, actions we take in preventing them, different management practices we're using to address the AMR issue, and then the stewardship programs that we have in place really led to a theme of continued improvement. So as you can see, this issue of being very complex and very widespread has always, all the stakeholders at the table really pushed to have a continued improvement on the topic. And so that we went from a human side and an animal health side and through all the species. We move into the third day on environment, the last leg of that three-legged stool of One Health. And if you notice, it kind of went from the large picture, the surface air water work done by the EPA and the big and slow type of research they're doing, down to the individual watersheds, down to the individual plant. So you can see that there are people evaluating the genetic phenotypic aspects of AMR and antibiotics throughout the whole ecosystem. So that it gives us another look at that three-legged stool of One Health. And just to move into the last piece of that, as all these activities start from you as an individual, expressing your values, to us as species groups and producers, and humans utilizing and using these valuable tools we have as antibiotics, up to global policy and actions that we covered in our last section today, where the science-based research driving policy and action and the amount of activity that's happened over the last 5 to 6 years is overwhelming. I mean, to have all these action plans in place, continually raising the bar on the issue is something that tells us the conversations and dialogues that started with NIA 10 years ago has brought to a big ballooned into something greater as preparing for this crisis around the world, for lack of a better term. So the building consensuses, coming to common languages and then shared actions is from a global standpoint is ballooning over the last 5 to 6 years. So we see this issue move forward on that. And then it kind of fits with the mission of NIAA, and that is the mission of NIAA is to provide a forum to facilitate and engage industry leaders and organizations to derive solutions on the most current issues in animal agriculture. So I feel that my involvement in this symposium for the last 10 years has helped animal agriculture be a valued part of the discussion. So much so when we look at things like a US champion to talk on the international stage about AMR, I hope that they can take into account having some animal agricultural influence on selecting that position. And I'm sure that will be part of that One Health approach on there, down to building the consensuses across those fields. And finally, I want to leave you with two statements. It was said in a symposium about 5 years ago that if you think you understand everything about antimicrobial resistance, it hasn't been properly explained to you. The complexity of this issue is so broad and immense that it'll be a continued effort going forward to address it. And lastly, everyone looks at this issue through their own lens. And so as the things that I pulled out of the symposium could be completely different than what you pulled out of the symposium. But I would hope it gave the opportunity to look at the antimicrobial resistance issue through a couple different lenses and brought more information to the table to help you understand the complexity of the issue, the policies and actions that can help drive these action plans, and really know that we all are in this with the same goal. We may just be talking different vernaculars and have to develop common languages. And part of this session today, although we weren't in person to work it out in the hallways and around the dinners and at the bar afterwards, is all part of this process to move and address this issue. And so I would hope that you could see from the last 3 days, it went from a personal values issue to the breadth and depth of a global issue, all within 3 days and 3 sessions. And we have some challenges and some other thank yous that we need to provide for the group. And I'll turn it over to my co-chair, Dr. Leah Dorman, to carry on with that.
Hey, Eric, thanks for that great summary. And I just want to thank everybody for attending. I want to thank our sponsors once again, as well as our planning committee. I do have just a few call to actions. So 3 things. First of all, fill out the survey. That's how we get better. That's how we know what we're doing. It gives us ideas for topics for next year or even at our annual meeting. So watch your inbox tomorrow for that survey, and please make copious responses because we do look at those and they do help us. So the second thing is, if you're not a member of the National Institute for Animal Agriculture, we would love to have you as a member. So I want to put that plug out there. You can check out the website at animalagriculture.org and become a member through that. And if you have any questions about the symposium, if you have questions about becoming a member, don't hesitate to ask any one of us. JJ, Morgan, I know are there from the staff. Eric and I would be happy to answer any of those questions as well. And the third thing I'm asking you to do is let's keep this dialogue going. Eric mentioned that earlier. Let's keep this dialogue going. And one of the things that I did last year was I want to issue a 3 in 30 challenge. My challenge to you is in the next 30 days, have 3 conversations with people outside of agriculture. And take the opportunity to tell the story of responsible use of antibiotics. Take some of the tips that we've learned from the communications piece and some of the information that you've learned in the days following that in the symposium, and get out there. 3 conversations in 30 days, you can do it. And thanks again for coming. I hope you have a great rest of your afternoon, and I hope to see all of you soon.
Yeah, we should probably be remiss if we didn't also thank the NIAA staff.
Thanks, Eric.
For making this run so smoothly, as well as our sponsors, everyone else, but they truly did make this go seamless, and we appreciate that. And again, thank you again, and we will see you down the road.